Good morning, delegates. I welcome you to MicroCon 2024, the 47th Annual Congress of the Indian Association of Medical Microbiologists, organized by the Department of Microbiology, Byramjee Government Medical College, Pune, in the cultural capital of Maharashtra. We have many workshops today, and we are delighted to have you join us for one of the most remarkable lineups of 18 pre-conference workshops at MicroCon 2024. Our pre-conference workshop at serial number 11, titled Advancing Clinical Microbiology with Illumina Next Generation Sequencing, is organized by the Department of Microbiology, Byramjee Government Medical College, Pune, and is coordinated by Dr. Rashmita, Assistant Professor of my department. The faculty will equip you with a thorough understanding of how Illumina Next Generation Sequencing, that is the NGS technology, can revolutionize clinical microbiology. They will offer hands-on wet lab experience and data analysis sessions, providing practical skill in integrate NGS into clinical practice. We encourage you to actively take part and understand the intricacies of this advanced technology that is bound to transform the future of modern medicine. Welcome again.
There might be clinical mutations, there might be a SNP which might be related with a particular disease. Okay? Now, it will also be able to understand the composition of microbiology or microbial composition in the particular samples. For example, you have a stool sample with you and you want to understand the diversity of microorganisms there. So you can do that by performing using NGS. And beside this, by using NGS, and you can also determine how genes are regulated. Okay, so we have this methylation sequencing, which helps to understand the methylation pattern in your genetic materials. That way, we may be able to understand how genes are regulated at the level of DNA. Now, at the level of RNA, you will be able to understand what are the various transcripts which are expressed in a sample. Okay. And what are the various fusions, DNA transcripts, isoforms of a particular genes which are available from a particular genes. Okay, so that can be understood at the level of RNA. Now, at the level of protein, you will be able to understand what are the various proteins which are expressed in a particular sample. For example, you have, let us say you have a cancer sample with you. You can even compare it with a normal sample and try to understand what are the various genes that is being expressed, various proteins that is being expressed, upregulated in the cancer sample, downregulated in the cancer sample. That way you may be able to understand the entire pathway which is working in the cancer sample. So that those things can be done. Now, why do most of the labs they are using NGS? Now, by using the power of genomics, or by using the NGS machine, it has various applications in different fields, in reproductive health, in genetic disease, in oncology, in research, infectious disease, drug discovery, and proteomics. So, why most of the labs they are using this NGS? Through NGS, we are trying to understand, or we are trying to enhance the discovery of genetic variations. It helps us more broader understanding of genomics. How it can be done by collecting qualitative information and how this qualitative information can be collected. So by NGS, we will be able to understand the mutations or the changes in the particular samples. Mutations or changes, for example, if you consider the SNPs, the insertion or deletion that is happening in the particular genetic materials, and how they are associated with the disease or cancer. So this can be one question which can be understood. Then you can even compare two samples together. You may be having a case and control samples, and you can compare the genetic makeup between these two samples. Besides this, let us say in recombinant DNA technology, in companies where they are working with genetic modification in microorganisms or in higher organisms, they can check whether the modifications were successful or not. Okay. For example, let us say they have cloned a particular genes and whether the cloning was successful or not. So this can be understood by utilizing NGS. Besides the qualitative information, NGS can also help you to collect quantitative information and understand the diversity, as I mentioned earlier, of microorganisms in the samples, which is also considered as a metagenomics application. In epigenetics, we can try to understand the methylation patterns in our gene at the level of our genetic materials, and this will also help how genes are regulated, gene expression are regulated. RNA sequencing, this will help you what are the various transcripts which are expressed in a sample. Okay. And resequencing, when you are particularly sequencing the organism samples again. For example, during COVID, initially the sequencing was done to understand the genome of the genome of your COVID virus. Later, we continue performing COVID genome sequencing to understand the various types of COVID variants which was coming into the society. So this was some basic idea about genomics and sequencing. Now let us jump into the Illumina sequencing overview. Here we will try to understand the workflow that is with Illumina sequencing and sequencing by synthesis chemistry that Illumina follows. So these are the basic workflow. So the first step in sequencing is to extract the genetic material. So it can, your genetic material or the nucleic acid can come from any kind of samples. It can be from microbiological samples. It can be from any kind of tissues, blood. Okay. So once your nucleic acid is extracted, the next thing is your library preparation. So what we do generally in library preparation. So today, throughout the workshop, we are going to focus on library preparation. So we will make you understand what are the various steps which are involved in library preparation. So in library preparation, what we do, we are making the genetic, your DNA or RNA samples compatible for sequencing over the NGS platform, Illumina NGS platform. So Illumina NGS platforms, these are short fragment sequencer, which means they only sequence small fragments of DNA or RNA. Okay? Now, during library preparation, we are fragmenting your genetic material into smaller pieces. We will be ligating certain adapters and making it compatible for sequencing over the NGS platform, Illumina NGS platform. Now, once your library is prepared, we are going to load over the NGS machines where clustering and sequencing will happen. Okay, so we will talk about this clustering and how sequencing will be done. Now, sequencing can be done in any one of the NGS platforms that is available with Illumina. Here in the diagram that I have shown, this is a Nova 6000 platform. So we also will discuss about the different platforms that is available. Now, once the sequencing is done, data will be generated, which will be in the form of a file known as FASTQ files. These FASTQ files will be run over a software. So today, in the second half session, we will also have a demonstration on how to perform the analysis. So we have different cloud-based platform, command line platform, where you can upload the FASTQ files, perform the analysis, follow the commands, perform the analysis, and you will be able to know the genetic variations that is present in a particular nucleic acid. Okay, for example, let us say you try to understand the genetic, what is the genetic variation in a particular gene. During analysis, what will happen is that particular sequence information will be aligned to a reference sample, and it will help you to understand the genetic variations. Now, in library preparation, as I mentioned, so let us see what actually is happening in library preparation. So, you will be supplied with a RNA or DNA with you, means what you are going to extract the nucleic acid, right? Now, the nucleic acid will be fragmented into smaller pieces. This can be done by enzymatic way or by using some kind of physical methods, or we generally use ovaries, which utilizes sound waves to fragment your genetic material into smaller pieces. Now, once they are fragmented, we are going to add adapters at both ends, which will make sure your DNA is able to sequence over the NGS platform, over the Illumina NGS platform. So this is a complete library. The structure of complete library looks like this. You have the DNA insert in the middle, which is supposed to be sequenced. Now, during library preparation, the adapters are added on the 5' end of your DNA insert or the 3' end of the DNA insert. Now, if we look into these adapters, they are divided into smaller segments, the P5 and P7. Okay. Now let us discuss about the function of this. The P5 and P7, Illumina NGS, they generally happens over a glass line, glass sandwich. Okay. So which is known as flow cells. And these flow cells, there are certain oligos which are ligated over the flow cell surfaces. This P5 and P7, they are complementary to these oligos over the flow cell surfaces. Okay. So during sequencing, what will happen? This library, they will be denatured and they will become single-stranded molecules. When we introduce it over to the system, the library molecules, they are going to ligate over the flow cell surface because we have this P5 and P7 which are complementary to the oligos that is present over the flow cell surface. Then we have index 1 and index 2. These are a kind of molecular barcodes. So index 1 and index 2, the sequence information of index 1 and index 2 are known to us. So every samples, they are going to have a unique combination of index 1 and index 2. Now when you have, let us say, multiple samples, let us say you have 96 samples and you want all of them to be sequenced together. The index 1 and index 2 information is going to help us segregate the data coming out from the NGS machine during analysis. Okay. Then you have RD1SP. This is primer binding site for sequencing the forward sequence. And then you have the RD2SP. This is a primer binding site which will help you to sequence the reverse sequence. Okay. If you have any doubt here, just let me know. I can explain better. Is it clear?
That 5' and 5' is that a mistake or?
No, it's not a mistake. Forward, reverse. 5' here it will, there will be 3' and then 5', 3'.
RT1 and RT2, can you please explain again what is the function of those two parts?
Which one?
RT1SP, RT2SP.
Now, in order to sequence the DNA insert in the forward direction and the reverse direction, because we have two strands of the nucleic acid, right? The forward strand and the reverse strand. Now, in order to sequence the forward strand, we need a primer. So, RD1SP is the read one. The forward sequence is known as a read one. Okay. So, the RD1SP is the read one sequencing primer binding site. The primers will come and bind here, and it will help in sequencing the read one or the forward sequence. Then RD2SP is the region where the primer for sequencing the reverse read or the read 2 will come and bind. So these are actually primer binding sites.
And index 1, index 2 for?
Index 1 and index 2, these are actually molecular barcodes.
Barcodes.
Yeah, so the sequence length of index 1 and index 2 could be 6 bases, 8 bases or 10 bases. The sequence information of this index 1 and index 2, they are known to us. Okay. So what will happen at the end of sequencing? Now when you pool multiple samples together, 96 samples at a time, each one of the samples, they are going to have a unique combination of index 1 and index 2. So the information of these barcodes is going to help you segregate the individual FASTQ files, okay, at the end of sequencing.
So index 1 and 2 are also part of the adapter region?
Yes.
P5 and P7?
Yes. Not P5 and P7, this completely together they are known as adapters.
Okay.
Just one example here. So you have multiple samples together. Here you can see 10 samples. After sequencing all the data, these are all the libraries, right? You are pulling all of them together. Now after sequencing, what will happen? How we are going to get separated? Because they have a unique combination of index 1 and index 2. So the data will be segregated and then depending on this information, from this information they will be segregated into individual files. Now once your library molecules are ready, you are going to denature the library. It will be separated into individual single strands and you are going to load over the NGS machine. Okay. So the first step that happens over the NGS machine is clustering. Okay. So what actually is clustering? Now when a single library molecules, obviously they are going to bind over the flow cell surface. They have this P5 and P7 which are complementary. right? When they bind on the flow cell surface, a single library molecules, they cannot emit light which can be captured by the optical system in the NGS machine. So in the NGS machine, we have this optical system or the camera. Okay. Now, single library molecules may know the intensity which is coming out from the single library molecules. It will be very weak that the camera is not able to capture them. So in clustering, we amplify the single library molecules into a multiple clonal clusters. So from one molecule, it will become 3,000, 4,000 molecules. Now the intensity is coming from a cluster of these clonal clusters. It will be so strong that it can be easily captured by the camera or present over NGS machines. Okay. Now, as I was talking about the flow cell. So flow cell is actually a glass sandwich. You have bottom surface as well as you have the top surface. Now, over these surfaces, you have oligos which are ligated over the flow cells, and these oligos are actually complementary to the P5 and P7 region of the library molecules. Now, when the library molecules are introduced, what will happen? They are obviously going to bind there on the flow cell because they are complementary. This is the flow cell architecture. So we have two types of flow cells. One is known as the random flow cell and another known as the pattern flow cell. Now, the difference here is in random flow cells, we cannot control where the library molecule is going to bind. It will bind randomly over the flow cell surface and the cluster is going to form randomly. We cannot define the size of the cluster as well. It can be large or small. However, in case of pattern flow cell, there are specific nanowells which is present over the pattern flow cell. So the oligos, they are present within these nanowells. Thereby, all the cluster is going to form within these nanowells only. So most of our high output or high throughput NGS platforms, starting from NextSeq 2000, then we have NovaSeq 6000, then we have NovaSeq X series. All of them have the pattern flow cell. However, the smaller platforms like the MiniSeq, then we have MySeq, then we have NextSeq 550, they have the random flow cells. By using the pattern flow cells, we are also able to generate much more data. That's why most of our high throughput platforms, they are utilizing the pattern flow cell. So let us see how clustering happens. We will just consider here a single library molecules, though there will be multiple library molecules and clustering will be happening throughout the flow cell surface.
You said multiple samples can be run on NGS.
Yes.
And if suppose in a single sample, multiple targets has to be seen. So what is advice? If multiple targets, like suppose stool sample has to be extracted and we have to run and see how much microbes are there in that stool sample, different types of. So can we run multiple samples and we can get multiple answers from the same sample in the single sample, how many microbes are there?
Yes. So let us say you have multiple genes to investigate it. Okay. So one cancer sample, whatever the genetic material that is coming from these cancer samples will be considered as a single sample only. Okay. There might be multiple genes, but all of them, they are going to have similar indexes. So that way we will be
Suppose to give an example, I have 10 cancer patients lying down with me and
yes.
I need to check all the 10 samples endogenous gut flora. So can I run all the 10 samples?
Yes, you can do that.
And then I can get an answer that patient 1 has this sort of flora and multiple organisms that we can check it out.
Yes, you can.
And then which flow should be used, that random flow cells or pattern flow cells?
It doesn't matter, you can use any one of the flow cells. It depends on which platform that you are using. Okay, so if you are using the lower platform that we have, it will use the random flow cell. If you have the high throughput platforms from Illumina, they are going to use the pattern flow cells only. So means the application or the output is not defined by the types of flow cells that you are using. Okay. So as I mentioned, now we are going to use indexes in order to differentiate the samples. For example, you have 10 samples, 10 fecal samples, let us say, from different patients. Now, patient 1, whatever the genetic material that you have extracted from the fecal samples, they are going to have a unique or single barcode information. Patient 2, they will be having a single different barcode information. Patient 3, they will be having a different barcode information. Based on this barcode information, after sequencing, you will be able to segregate the data. So data will come out as a FASTQ files. Okay, so patient 1, different FASTQ file, patient 2, different FASTQ file. But during analysis, you will be able to differentiate what is the microbial diversity between patient 1 and patient 2. That will happen during analysis. Similar also goes with the cancer samples also. So in you might be having 100 genes panel. Okay, we normally goes with gene panels. In cancer, we have like different solutions are also available with us. So patient 1, let us say you have 2 patients and you want to check the difference in the your genetic variations among these 100 genes. Okay, between patient 1 and patient 2. You can perform NGS. Patient 1, what will happen? You are going to have a single index combination. Patient 2 also, you are going to have a different index combination. After sequencing, both of the data will be segregated, but the genetic variation in every individual gene will be understood during analysis. You are going to align your sequence with a reference genome, and the genetic variation can be determined. So that will be done during analysis. I hope that is fine. So as I mentioned, we will be just now focusing on a single library molecules. Though multiple library molecules will be undergoing the same process. Now, after the library preparation is done, you are going to denature the library molecules. Okay, introduce over the flow cell surface. These library molecules, as I mentioned, they are they have this oligos E5 and E7 molecules, which are complementary to the oligos over the flow cell surface. You can see these are different oligos which are ligated over the flow cell surface. The library molecules, they are going to come and bind because they are complementary to each other. Now, by utilizing these oligos as a primer, there will be chain extension. Okay. So the first thing is here you need to understand is all the requirement, the enzymes, the nucleotides, okay, all the buffers that is required, it will be provided by the machine because we have a cartridge which has individual reagents, and whatever the reagent which is required for this extension will be provided through the NGS machines. Fine. So chain extension will happen from the single library molecules. Now it will become double-stranded molecules. You have the original template as well as the newly synthesized strain. This newly synthesized strain, these are actually ligated or is covalently linked to the oligos over the flow cell surface. The next step is to denature them, and this original strain will be discarded. So denature and the original strain is discarded. Now you have the newly synthesized strain which is covalently linked to the oligos over the flow cell surface. Okay. Now this was one slide here. This one that how individual library molecules they are randomly binding over the flow cell surface. Okay. Since this is a single-stranded library molecules, and when we are flowing all our reagents over the flow cell surface, they have a tendency to bend down. Okay, they are continuously moving, and they have a tendency to bend down. So they will bend down, and because they also have a complementary sequence for the other end, they will bind with the oligos with on the flow cell surface. There will be extension again, and this results into a double-stranded breech-like structure. So here that's why we call it breech amplifications. Okay. Now this breech amplification, this breech-like structure will be denatured again, and at the end you will have two library molecules. One of them is a reverse strain and another is the forward strain. Okay. One of them is the reverse strain, another one is the reverse strain. So this breech amplification will continue, resulting in the formation of clusters. Now in a cluster you have both the forward as well as the reverse strains. Got it? Because we cannot sequence both the forward and reverse strain together. Why? Because the signals will be a kind of mixture. Okay? So we need to sequence the forward strain first, then the reverse strains afterwards. In order to do this, we are going to cleave the reverse strands. After cleavage, you will only have the forward strands ready for sequencing. So now the forward strands are ready for sequencing. You are going to provide the primers which are required for the forward strand sequencing or the read one sequencing. The primer will come and bind to the read one sequencing primer binding site, and your forward or the read one sequence, they are now ready for sequencing. And this sequencing can be done in any of the NGS platform, which is provided by Illumina. You have the MySeq, MiniSeq, iSeq. These are low throughput platform. Then you have the Next 550, Next 1000, 2000. In high throughput platform, we have the NovaSeq 6000 and NovaSeq X Plus. Okay, so these are the different series of platform that we have. Now let us try to understand the basic chemistry behind the sequencing by synthesis. Now once your sequence are ready for sequencing, what will happen? The machine is going to introduce the nucleotides, the 4 nucleotides. So all these 4 nucleotides, they are labeled with different fluorescent dyes. Okay. Beside this fluorescent dye, they are also labeled with another chain terminator molecules, which means, if we consider this example, you have the library molecules ready for sequencing. You have the primers which has come and bind there. Now the system is going to introduce the 4 nucleotides together. Now depending on what is the complementary here, one of the nucleotide molecules is going to bind. Okay. So because these nucleotide molecules, they are labeled with number one, there is your fluorescent molecules, another is a chain terminator molecule, which means wherever it binds, there won't be any further extension. Okay. So there won't be any further extension. In the next step, the fluorescent molecules is excited, and the camera is going to capture the wavelength of radiation that is coming out from these fluorescent molecules. Okay. Once the camera captures the information, the chain terminator molecules as well as the fluorescent molecules will be cleaved off. It will be removed. Now the molecules or the library molecules is ready for the next molecules again. Okay. So in one cycle, what will happen? There will be incorporation of the nucleotide molecules. Then there will be excitation, and the camera will take the picture. Then there will be a cleavage. This is happening in one cycle. So let us say you have a 150 size DNA fragment. Now, in order to sequence in 150 DNA bases, you need to perform 150 cycles of sequencing. So in every cycle, these steps are repeated, where there is incorporation, excitation, taking of the picture, then removal of the fluorescent molecules. Okay. So in every cycle, the camera is going to take 4 pictures in different wavelengths, because these fluorescent molecules, when they emit radiation, they are emitting in different wavelengths. So let us say G, they are radiating in blue wavelength. T, it is radiating in the green wavelength. A, it's radiating emission in the yellow wavelength, and C, radiating emission in the red wavelength. Okay, so 4 pictures will be taken in 4 different wavelengths. So out of this 4 pictures, in only one of the picture the image will come, and from there we will understand that it was whether the particular nucleotide which was incorporated was G, or whether it was T, A, or C. Okay. So in every cycle, 4 pictures and 1 nucleotide will be identified. I hope this is clear. So this is how the forward sequencing is done. The same will happen in the reverse sequencing also. So when we want to sequence the read 2 or the reverse sequence, we call it pair-end sequencing. Okay. So single end means you are just sequencing there are 2 terminologies we generally use. One is single-end sequencing, another is pair-end sequencing. So in single-end sequencing, we are just sequencing the forward bridge. When we say pair-end sequencing, we are sequencing the reverse bridge as well. So when we perform reverse bridge sequencing or the pair-end sequencing, it increases the data that is being generated, and it also increases the confidence of our sequencing. Okay, so most of the time we generally prefer to perform the pair-end sequencing, but there are certain applications where we just go for single-end sequencing. Applications like where you try to understand the transcript or the gene expression level, the transcript level. We normally sometimes go only for a single-end sequencing because we are just counting the number of transcript that is being expressed. So we don't have to do with the actual sequence of nucleotides there. So certain applications are like this where single-end sequencing is recommended. So this after the forward read or readward has been sequenced, you are going to have a product like this where there is the oligos which is ligated and the sequence molecules. Okay, so you need to denature this. The system is going to denature, and the system is again going to follow the same bridge amplification. So at the end of bridge amplification, you will be having two library molecules, the same forward as well as the reverse. Because we have completed the forward sequencing, now the next thing to do is the reverse sequencing or the sequencing of the rate 2. So we are going to clip off the forward strand. Now the reverse strand is ready for sequencing, and for that we will provide the primers which is required for the reverse strand sequencing. Okay. Now sequencing of the reverse strand is also going to happen with the same method, sequencing by synthesis technology. It will happen. It will follow the same method. Now what we have discussed so far was about the read 1 and read 2 only. Besides this, you also need to collect the information of the index 1 as well as index 2. So index 1 and index 2, you need to sequence them. They are also going to follow the same SBS method or sequencing by synthesis technology. How it is done? So once your read 1 sequencing is done, complete, the primers required for sequencing the index 1, or we call it I7 index, it will come and bind and it will help in sequencing the index 1. Okay? Then there is a pair-end turnaround. Pair-end turnaround is nothing but formation of the cluster here. Just now we have discussed clustering, right? So clustering was happening 2 times. One, when there was sequencing required for the read 1, another during sequencing of the read 2. Okay? So pair-end turnaround or the reclustering will happen. So before sequencing the read 2, the index 2 are sequenced. Okay, primers required for sequencing the index 2 will be provided and it will be sequenced. Then at the end the read 2 will be sequenced. So these all primers, they are provided in a cartridge which is provided by Illumina. Okay, so all the reagents, all the enzymes, all the primers will come inside a cartridge. So once sequencing is done, your data will be generated as a FASTQ files, and we will perform data analysis by various methods. Various software tools are available. Freely available software also there in the market, and Illumina also does have various cloud platforms where you can just upload the data and they will have perform the sequencing. Data analysis. Okay, so analysis, if we look into it, it can be divided into primary analysis, secondary analysis, and the data interpretation. In primary analysis, it's happening over the machine itself, where the pictures or the photograph that was taken, all of them will be analyzed, and the system is going to extract the information of nucleotides in every cycles. Okay. So once the primary extraction is done, the next thing in your secondary analysis is after the primary analysis, your FASTQ file will be generated. In secondary analysis, the FASTQ files, they are going to be aligned to a reference genome, and there will be a comparison between the sequence of your genetic material as well as the reference genome, and the genetic variation will be identified. Now, once genetic variation is identified, during interpretation, it will be annotated, which is going to give you insights whether it is related to a particular, whether the genetic variation is related to a particular disease, or what kind of, in case of, let us say, disease like cancer, what kind of drugs should be targeted to that particular individuals. Those information can be like determined after your sequencing is being done. Okay, so there are different tools and different softwares which will help you to perform that. Even in Illumina also we have ICI, ICA, Illumina Connected Insights, which helps to interpret those kind of data.
So after the clustering is done based on index 1 and index 2, FASTQ file is exactly, it's like the terminology exactly is for what? Because clustering has happened. After that, I jumped into right into the FASTQ.
Okay. Clustering. Let me explain, let me like tell you where so your library molecules are ready, right? They have been introduced into the flow cell surface. Clustering will happen there. Okay. Now clusters are being generated. Now once the cluster are generated, in the cluster, as I mentioned earlier, you are going to have the read 1 as well as the read 2. Okay. Both forward as well as reverse read. So during read 1 sequencing,
Why are we sequencing both the forward and reverse? Because anyway we are sequencing the single stranded fragment. So why forward and why reverse?
As I mentioned earlier, by sequencing the forward and reverse read, you also get higher number of data, and you also have much more confidence over the data, over sequencing information.
So when we say the depth of the data increases, is it this part?
Yeah, this is this part. I mean, you can increase the depth of the particular, the sequencing depth can be increased by performing both the sequencing of the read 1 as well as read 2.
So what is the difference when you know the depth is less versus when the depth is more? Like it's of the same target.
Yes.
So when we are taking that, going on sequencing that same target again.
So during sequencing, we provide, say, if you want a higher number of depth in a particular sample, okay? So during sequencing, what we do, we increase the loading concentration of that particular samples, okay? So let us say you have two samples, sample A and B. You want more samples, or you want more depth or more data of sample A compared to sample B. So when we load the library molecules over your machine, we are going to have 60% of your loading concentration given to your sample A, and another 40% of your loading concentration given to sample B. That's how we increase the data generated from a particular sequencing, during sequencing. Let us say, see, every flow cells, we have a data output. Okay, which may be like 120 GB, let us say, 120 gigabases, 120 gigabases, we call it 120 gigabases. Now, this 120 gigabases can be distributed equally in among samples, or you may give more data to a particular samples. Okay. Now, when you require more data, means more depth means more data. If you need more depth, you need more data from a particular samples.
No, sorry to interrupt. I do not put the question properly. What I meant to understand is, by increasing the depth of the data, how is my data becoming better? Because it's the same sequence that I'm going on, doing over and over again. So is there a chance of missing out on something if I do it lesser number of times versus when I do it more number of times?
When you perform more number of times, you will be able to identify certain novel mutation as well, novel genetic variations also.
Okay, so in sequencing,
yeah, there are various. See, when we perform a sequencing, certain genes, the genetic variation may be very rare. Then you need to give more data to identify that particular variation.
So yeah, I less data, so your confidence is less. So you are looking for some targets, just B3 or yes, when you are looking for the de novo sequence, that is sequence and reference you have, then you go for the depth is small. Suppose the 100 depth is there, so 99 times there is A, this stated with detect machine A same, but time is T. Computation level is high, so it's 99% A, so it should be A1.
So it's a kind of statistic for the machine.
Yeah.
So, see, as I mentioned, means when you increase the depth, there is higher chances of identifying novel variants also. There might be like genetic mutations, okay, which we are trying to identify. But at a lower depth, you may not be able to identify the genetic variation. But when we provide a higher depth, the rare variation may also be identified. So it's up to the user, means how much depth or should be needed for a particular sample or a particular genetic variation to be identified. So depending on that, we choose how much depth to be given. So by performing the pairing sequencing, we can increase the depth and we can also increase the confidence of that particular information that we are getting. Okay, so yeah. So as this is a workflow, so should I like explain this again, ma'am?
No, I got it.
Got it? So these are the various platforms that we have. I see this is the lowest throughput machine which can generate up to 1 gigabases of data. My MiniSeq which can generate up to 7.5 gigabases of data. Then MySeq it can generate up to 15 gigabases of data. So this here GB is gigabases, okay, no gigabytes, right? So when we define human genome, we define the size of human genome as 3.2 gigabases. Okay. Then you have MSeq 550 which can generate up to 120 gigabases of data. NextSeq 2000, 1000 which can generate, now it is able to generate up to 520 gigabases of data. Then you have NovaSeq which is able to generate 6,000 gigabases or 6,000 TB of data, 6 TB of data. So beside this, we also have NovaSeq X series which are able to generate up to 16,000 gigabases of data. Okay, so
in what time, what workflows and what?
Yeah, I'll come into that next slide.
This amount of generation of gigabases data will also be surrounded with what we are looking for. If it is micro.
Yeah, I'll come into that as well.
Which among this is the ideal, or which among this might be the least required? At least this should be there for us to know micro sequence.
Now see, for microbes, most of the microbes, now let us go into the next slide and it will give you an idea. Now these are the different sequencing platforms and the key applications overview. Okay, so let us say you have large whole genome sequencing, for example, human genome sequencing. When we say human genome, the genome size is actually around 3.2 gigabases. Now in order to have a proper, you need to perform at least 30x, yani 30 times sequencing of a particular nucleotide in order to understand the genetic variation in a human genome, which means 3.2 GB multiplied by 30. That will give you around 90 gigabases, or let us say give or take 100 gigabases of data. Okay, so every human genome, in order to perform NGS, you require to give a sample around 100 GB of data per sample. Okay. Now let us say you have the microbes that are considered an E. coli or bacteria which might be having around 6.5 MB of data only. Okay, 6.5 MB is quite small. Now if you multiply 6.5 by 30x also, even for 100x also, what you are going to have? You are going to have just 650 MB, which is even less than 1 GB, right? Now in those scenarios, if we compare the high throughput machines, you can see here 120 gigabases. You will be able to sequence only one human genome. Okay. Now, if we consider this 330, maybe around 3 human genomes will come, because one human genome you require how much? Around 100 GB of data is required. If we consider this NovaSeq 6000, this may help you to sequence around 60 samples at a time. Okay, 60 samples in NovaSeq. We have two sites, site A and site B. Side A can see this can generate 3000 GB of data. Side B can also generate 3000 of GBs of data. Both of them can be run simultaneously. Totally they can generate 6000. That's why it is written as 6000 GB. So in 6000 GB you will be able to sequence 60 human genomes. But this platform we will not recommend it for sequencing a smaller organism like bacteria, because bacterial genome is just 6.5 MB. And even if you sequence it for 100 times, how many data will be there? Only 650 MB is required. Now, in that scenario, smaller platforms like iSeq, MiniSeq, or MySeq, they may be more suitable. Okay, you can see MySeq, they can generate up to 15 GB of data. Okay, let us say you have an organism, small organism, which, for which the data requirement is around 1 GB per sample. So you will be able to sequence around 15 samples here. Okay? So at the time, it may not be possible, you may not be having 120 microbial samples. Okay? In NextSeq, you can also do that, but you need to have a larger number of samples, 120 samples with you. If you consider the NextSeq 2000, how many samples will be there? You need to have around 350 samples in order to set up one run. Okay, so sometimes this may not be like a suitable platform, because at the time you might not be having 300, 400 samples. Okay. But it can be done. It can be done. Okay, all this sequence, all the machine, the higher platforms can also be utilized for sequencing the microbes genomes also. That can be done. You can even what you call, like, let us say you have in this, if you consider 330 GB, let us say you have 3 human samples, right? Those 3 human samples, how many data will consume? Around 300 GB data will be consumed. Remaining 30 is left behind, right? So this 30 GB can be utilized for sequencing your microbial samples. You can pull them together and perform the sequence. Okay, that is not an issue. Likewise, in the higher level also, the same thing can be
So
done.
MiniSeq can be still be ideal with the least number of samples also. Say MiniSeq system. What would be the sample minimum requirement for MiniSeq to be effectively utilized?
Yeah, MiniSeq, MySeq can be effectively used. This is the highest output that I have given here. We also have flow cells, which comes with the low output. I'll just show you in the coming slides. Now, as I mentioned, these are the major applications you can see here.
Can move back to slide. This one, so 1.2 gigabase that I got. The other points, 4 million, 2 into 150, what rate?
This is 4 million means they can generate 4 million clusters over the flow cell surface. Okay? 4 million clusters, 4 million, one cluster means one read, right? You are going to get information on one read. So 4 million reads, 4 million clusters information you will be able to generate. This will become clear when we go to the upcoming slides. I will also explain it here. Now here, this is a major application overview. You can see here.
One question. So for microbial genomics, you're saying that more amount of data is adequate. But for the same microbes, if we want to, like for research purpose, RNA sequencing, transcriptome kind of work, would it require a higher level of the sequencing?
Yes. So ideal platform, if you want to go for WGS as well as transcriptome, you can utilize this NextSeq 1000 2000. They have a minimum data output of 30 GB to have a maximum data output of around 540 GB. Okay, so this will be one of the ideal platform which is offered by Illumina.
So for 30 GB, what is the approximate million reads per sample?
Then we come to the next slide, because we also have a slide for every one of the platforms. Now this is the major application as mentioned. You can see high throughput, mid throughput. In high throughput, Nova 6000 X plus they are coming up. You have the whole genome sequencing application. Even the WGS of smaller organism can be done here. As I mentioned, you can do samples, human samples with microbial samples. You can perform the sequencing here. Exome sequencing. These are sequencing only the coding region of your genome. Then you have targeted gene sequencing. For example, you have a panel of genes and you want to understand panel of gene related to cancer. Okay, so So you can target sequencing can be done. Whole transcriptome, this is the total RNA sequencing. Okay. Or then you have the gene expression profiling, which is also known as the mRNA sequencing. Then you have targeted gene expression profiling, only specific genes of your interest and how they are expressing this chemistry. So these are how high throughput platform. Even mid throughput, the various applications as you can see here. However, in low throughput, certain applications are limited. You can see here, large whole genome sequencing, like the human genome, cannot be done on a mid throughput because the data requirement of human genome, as I mentioned, is about 100 GB, right? But you cannot do the human genome sequencing over a MySeq platform because MySeq platform is just having a maximum of 15 GB data output. Okay, so some applications cannot be done over low throughput. Even the exome sequencing per sample data requirement is around 8 GB. Okay, so it's not possible, or we do not recommend doing over the LoRa platform. Whole transcriptomes, it is also limited over the LoRa platform because the maximum data requirement, data outputs of a MySeq is 50 GB only, while it has, if you consider a million reads, MySeq is having maximum of 25 million reads. Okay, 25 million cluster can be generated. While in case of a transcriptome application sequencing, you require a minimum data of 50 up to around 50 million reads, total RNA sequencing. So it's not possible to perform transcriptome total RNA over a MySeq platform. Fine. Now if we go into which one of the specification, the I6 system, the lowest throughput machine, it can generate a maximum of 1 to 1.2 GB data, 4 million clusters, right? Runtime can differ from 9 to 9 hours. Okay? 19 hours. Then this is the total read length which can be sequenced. Now when we say read length, different configurations are there. It's 1 into 36, 1 into 50, 1 into 75, which means you are sequencing only read 1, okay, or the single end sequencing. 36 means it is sequencing only 36 bases. 50 means 50 bases, 75 means 75 bases. So this is single end sequencing. We have different flow cells which are able to perform single end sequencing, and they can sequence 35, 36, 50 and 75 bases. Now if you see here 2 cross 75 and 2 cross 150, which means they are able to sequence both read 1 as well as read 2. So when we do this, it is going to sequence 75 bases of read 1 and 75 bases of read 2. Likewise, 2 cross 150, this means it is sequencing 150 bases of read 1 and 150 bases of read 2. Okay. So all of them, they are able to generate 4 million clusters. Now 2 cross 150 gives you around 300 cycles, right? So 300 multiplied by 4 million, it gives you 1.2 GB of data. This is how we calculate the total data output of a particular process. Okay. 300 cycles, which is like forward 150, reverse 150. Your total is 300, right? 300 multiplied by 4 million cluster, total data output is going to be 1.2 GB of data, right? Now, it's up to you how you pull the samples together. Your data requirement might be, let us say, 600 MB. In that scenario, you will be able to run only 2 samples. 2 GB is like 600 MB plus 600 MB is going to be only 1.2 GB. Right? Now, let us say your data requirement is 1 million reads per sample. Then you will be able to run 4 samples. Okay? So, depending on your application, the library preparation or the application is going to define what is the data requirement for the particular sample. The data requirement may be defined in terms of million reads or may be defined in terms of gigabases. Okay, so it's up to the application. For example, in transcriptome or the total RNA sequencing type of application, the data requirement is defined as in terms of million reads. If we are going for example, human total RNA sequencing, we define the data requirement as 50 million reads per sample. Okay. But in certain applications like whole genome sequencing, data requirement is defined in terms of gigabases. Human genome sequencing will require around 100 GBs of data per sample. Okay. So the application will give you an idea about the data requirement, whether it is in terms of million bits or whether it is in terms of your gigabases, right? So these are some of the basic applications which can be done in your ISIC platform. So I'm not going to go through because I have shared one slide and I will also be sharing this slide so you can have a look around after the workshop. The next one is our mini-SIG platform, which can generate up to 7.5 GB of data, starting from 1.9 to 7.5 up to 25 million clusters, and it can it can have 150 PRN is 2 cores 75, and it can also have single and 75, 2 cores 75, then 2 cores 150. So these are the different flow cell options which is available.
Sorry for a non-academic question. Can you tell us how costly is this?
Price-wise, our sales team will give you better answer on this.
No, it's just for an idea.
This machine, if you talk about this machine, I'm just giving a rough estimate because I'm not actually sure about the price. It may come around 75 to 80 lakhs, machine itself. ISIC machine, the ISIC that I showed earlier, they are much cheaper, probably around 25 to 30 lakhs.
I have one question. You are mentioning all this specification, how many images and how many images of data that one machine can read. This is based on machine or this is based on the flow cell or this is based on combining machine and flow cell?
This is based on the flow cells.
So can we use flow cell or
This is based on the machine as well as the flow cells. We can because this machine, they have only specific flow cells which is specific for this machines only. Okay, we cannot use the flow cell for Novacell here. So each of the machine, they have their own flow cells, and that's how we define the data output. Okay. So we cannot like use the flow cell of mini-SIG and ISIG. Understand the diversity of organism. So in this scenario, we define the data requirement at this minimum of 50 million bits. So for every application, we have a data requirement. And how these data requirements help to answer your question. A minimum cutoff is there. So when you
will provide or you will search the web and
you are going to, because this is going to be your research questions only. So there will be ample of literature which will be available on the website. So you can see how people are following, what is the minimum data requirement. We do have certain recommendations, but sometimes user always not follow those recommendations. Let us say, as I mentioned, minimum data requirement for the biodiversity analysis of fecal sample is 50 million bits, right? But certain, depending on the questions, depending on your queries, this data requirement may be reduced to 25, 30 millions, 40 millions. It's up to the user. But what we generally follow is we give the minimum recommendations. So we, in the first, if you are doing it for the first time, we will suggest to follow the minimum recommendations and perform your data analysis. From there, you will get an idea whether there is able to answer your question. If there is able to answer your question, then you can reduce it next time to 40 minutes. If your answer is not, the question is not answered, you increase the data requirement again up to 100 minutes. It's up to the user. So now this is the BISIC. So we have different flow cell types here. Nano flow cells which can generate 1 million reads. Then you have another flow cell, micro flow cell, which can generate 4 million reads. The standard B2 flow cells which can generate 15 millions, and the B3 flow cell which can generate up to 25 millions. Okay. So this 25 million, they can generate up to data of around 15 GB. Okay. If you multiply 600 cycles with 25, it will give you roughly around 50 GB of data. Okay. So this is how we calculate the data in every process. If we talk about NextSeq 550, this is the specifications. We have two types of process. One is high output and another is mid output. This mid output can generate up to 40 GB of data. They can generate 130 million clusters. While your high output, it can generate 120 GB of data and 400 million clusters. You can see here 400 million, which means, for example, certain application write total RNA sequencing. If the data requirement per sample is 50 millions, you can run around 8 samples here. You can perform 8 total RNA sequencing over the high output flow cells. Okay. So that is how we calculate number of samples that can be pulled together or sequenced together. NextSeq 2000 new flow cell has come, which is known as P4 flow cells, and that can generate around 540 GB of data. So until few months back, we were having P3 flow cells, which can have a maximum data generation of 360. Minimum data generation is 10 GB here. So this is one of the versatile NGS machine where you can be utilized to perform large organism sequencing up to a very high throughput sequencing. Because you can see here, you can generate up to 10 GB of data to a maximum of 550 GB of data. Okay. So this is one of the versatile platforms. And these are some of the applications which can be done on this NextSeq 1000-2000 system. Exome sequencing in P3, as I mentioned, exome per data requirement is around 7 GB, 7.5 to 8 GB. 48 exome sequencing can be done together at a time in the NextSeq 2000 P3 flow cells. Okay. While in P2 flow cell type, you will be able to pull 16 samples together. Okay. Now, if you talk about mRNA sequencing, in mRNA sequencing, the data requirement is around 25 million reach over. Okay. So you will be able to perform 48 samples together over the P3 NextSeq 2000 flow cells. And NovaSeq, one of the high throughput machines, they can generate at a time if you are performing sequencing on side A and side B, as I mentioned earlier. They have two sides, side A and side B. If you perform sequencing, they will be able to generate 6,000 GB of data. These are the flow cell combinations that we have. We have SP, S1, S2, and S4.
I have a question here. In 2 slides back, we showed that the 2000 model
Yeah.
is NextSeq 2000 model. So it can, for like mRNA sequencing, 48 samples can be run together, or exome sequencing 48 samples. Does that mean that until and unless you pull all 48 samples, then only you can do one sequencing run? Like for samples, this is the problem. Now the recent,
so let us say if you have a large number of samples, you have the option to run on P2 flow cell, right? 16 samples together. Like your samples are
शांत
It's your question, answer.
Same problem is there, like for samples also, yeah, so we have an in-house sample sequencer. We also have this thing, next gen sequencer, but it is mainly used by the genomics people. We don't get a chance to do microbial sequencing there. So for samples, this is the problem. So at the latest sequencer that has been provided from Fisher Scientific. So, at a time you have to fill all 24, minimum 24 wells have to be filled for the sequencer to run. Suppose I have only, even if I take forward and reverse both are not required for identification. If I have 16 wells to fill up, so the rest of the 8 wells, I am wasting the high dye and the polymers, buffers, everything just to complete the run. So I just want to know what is the situation with you all on
You need to have that amount of samples at least. You need to perform the because 16 samples here. If you let us say like you don't need one example is like you don't need to go for 48 samples. If you have 16 samples, you can have a choice of the flow cell type. Less sample means lower flow cell you can utilize. Depending on that, you can also do. Now let us say you only have a limited flow cell. You have a P2 flow cell with you. Only you are just having P2 flow cells.
And our solution to this, we were also running similar situations for other laboratory tests. And in a medical college, naturally, reagents purchase is budgeting for that is always a problem. So what we tried to do was, for maximizing the use of the facility available, we used to inform similar sister laboratories elsewhere in the town or nearby.
Yes.
And pool the samples from various sources. Give them code numbers according to the sources that we received samples, and her difficulty is genuine. You can't waste the reagents just because the number of wells are running short. And then you make up whatever 96 wells or whatever the size of the requirement, fill up with the samples received from elsewhere, and share the reporting at the time when your reports are ready with data analysis.
Yes, sir.
So that will curtail on the expenses of others also in procuring the machines and the reagents. At the same time, it becomes like a reference center for the nearby places for answering this type of sequencing data analysis. So this can be a solution.
Yeah, there is a solution.
There is a solution.
There is how because we have service providers also.
Yes.
In the country. So service provider they have a single unit, but as I have just asked the similar question here. The service provider, what they do multiple samples, multiple applications together. Okay, and perform the sequencing. Let us say you have a P3 flow cells, and you have you can utilize, you can pool multiple applications. Like exome can be pooled together with another application like transcriptome. Then it can be pooled with another application like WGS. So if you have multiple applications, and you can pool them together, and you can, because data requirement will be different, right? So you can maximize the utilization of this particular type of process. That can be like, that is what the service provider has been doing.
I think even for microbiological testing, would have samples for fungus, would have samples for
Yes,
bacteria,
yes.
some samples for viruses, and accordingly reference.
Yeah, because
Yeah.
different like sir has mentioned, virus, bacteria, other organisms, small organisms.
Fill up
You
the
can
requirement.
prepare the library. Each of the samples is going to have their own index combination, different barcodes. Put all them together, fill up the data requirement, and you can proceed with the.
I think that should be.
That is how people are also doing. Service provider, they are doing that.
Private labs are doing like that, but when as individual institutions like us, it's a problem. And also, I wanted to understand, is there after the nucleotide extraction has happened? I also wanted to understand, after the nucleotide extraction has happened, is there a chance of cross-contamination between microbial genome and the non-microbial or the human genome? Because our genomics people are always very worried that,
Obviously,
we should not be
like if you are extracting the nucleic acid, and if you want from a human sample, and when you want to go for understanding the information of that particular microorganism only, there will be human genetic material will also be coming up, right? We call it cross contamination.
That part we understand, but
should people like
so that nucleotide extraction facility can be kept separate, microbial and genomics. That part is okay. But after the nucleotide extraction has happened, can we do the sequencing run just to pull up the samples and so that you know the minimum number of sample requirement it is fulfilled?
That can be
The
done.
microbial and the human genomic sample.
That can be done. That can be pulled together because the samples, once the libraries are prepared, they are going to have their individual indexes now. So if you put them even the human and microbial samples together, at the end of sequencing, the data will be segregated depending on index information. So you will get your own microbial information, and somebody else is going to get their own human genome information. That can be done.
Thank you.
So this was the various flow cell type which is available from 6000. I am not going to go in details here. Let us jump into the recommended key applications. As I mentioned, because this is a high throughput machines, the human genome, exome and transcriptome, the numbers of samples that can be run together. In S4, approximately up to 24. We have seen people who have utilized up to 30 samples in this S4, so that can also be done. Exome up to 250, transcriptome up to 200. Okay, so this is the maximum number of samples that can be put together and that can be spread over a 6000 platform. So these are different flow cell type: SP, S1, S2, and S4. Number of samples for these three different applications. Now this is the NovaSeq X series. So in X series we have two types of platform: NovaSeq X and NovaSeq X Plus. The only difference is in NovaSeq X, they have only a single site and it can generate up to maximum of 8,000 gigabases of data, while in NovaSeq X Plus, they have site A as well as site B, and together they can generate up to 60,000 gigabases of data. Then we just jump into the applications because we are short of time. Now this is the key applications which can be run over the NovaSeq X series. We have three types of flow cell here: 1.5 billion, 10 B, and 25 B. Means it can generate 1.5 billion clusters. Okay, 25 billion clusters, and maximum of up to 750 exomes can be accommodated in this particular flow cells, and up to 520 transcriptome or total RNA can be done here. Okay. If required, I will have to understand how to calculate the number of samples in the flow cells. Okay, later on I can do that. Now define the key applications, because we have reached in different fields in oncology, even in food industry, agriculture, health, and infectious disease. All in these key areas, your genomics can be used, and it will help you to understand the biological questions in every field. Now let us see the sequencing application. Let us say you have an organism. It can be, your materials can be like, it can be your DNA or it can be at the level of RNA. Now in DNA, you can have targeted or whole genome sequences. When I say targeted, it can be exome or it can be a group of genes, or we call it a panel, or it can be just a single gene or single amplicon. When we say single amplicon, example is your 16S ribosomal RNA sequencing. 16S ribosomal RNA sequencing is a type of application which also helps you to understand the diversity of organism in a given sample. Then at the level of RNA, you can understand how genes are regulated by understanding your mRNA transcript, your microRNA, small RNA, total RNA, and at the level of DNA methylation, you can even understand how genes are regulated. Different applications, different methods can be applied in using your next generation sequencing. One key application of NGS is during the COVID time, which help us to identify the COVID virus genome and the variants that comes later on. So today we will be preparing the COVID virus genome. We will be following the library preparation protocol of the COVID virus genome. And in the afternoon, we will have a demonstration of how to analyze the COVID virus genome. And it will give you an idea how the variants are detected. So later on, once during the pandemic, once your COVID genome was sequenced, it also helped us to design the vaccines, right? In order to design vaccine, you require the genomic information. Genome information was identified, and later on, the vaccine information was able to, like, we were able to design the vaccine against the COVID, and we were able to control the pandemic because of that. And another one application, major application is that we also have a panel. I'm just giving some certain examples. We have different types of panels, and in the second session also, one of the product managers from Illumina, he is going to highlight the different applications of microbiology and NGS. Okay, so the panel, one example of panel is the respiratory virus oligo panel. So this panel can identify or detect up to 40 viruses, which may be directly or indirectly infecting the respiratory tract. Okay. So more information, you need to just visit the Illumina website. We have specific information related to cancer research, conflict disease research, microbiology, and reproductive health. Just go to the link that is given here. I'm going to share the slide to you so that you can explore more. Previously also I have provided certain links over the WhatsApp group. You just need to explore that. The Illumina school, the link that I have given, is registered there. You will be able to explore like different applications there as well. We have YouTube page also where more information about the different platforms are also given, and certain webinars are there depending for the focus of specific applications. You can explore that.
There are good videos also. Actually, if you display the video, you will be benefited.
Okay.
That the WhatsApp message that you have sent video, particularly the sequence and the cleavage, that will enlighten all the audience here.
Yes, sir. I might have.
My request, that's all.
Yes, sir. I might have a. How is the internet connection?
Can I give you on my mobile? I have on the mobile. The video is there.
It's there, sir. Sample preparation to have the extract your nucleic acid. This is library preparation, one of library preparation method. Fragmentation is by enzymatic method. You are fragmenting your nucleic acid here. Then you have the DNA insert. The adapters are ligated on both ends. Now this will give you the complete library at the end of this. So once the library is completion done, then you have clustering that will be going on over the flow cell surface. You can see the flow cell surface. Oligos are there over the flow cell surface. Now over this oligos, the library molecules, after the initiation, it will become single-stranded. They will go and bind here. Now it has bound. This will then be amplified, and you are going to have a double-stranded molecules. The modular strand is denatured and they are removed. Pre-amplification. So this is how pre-amplification is being done. So you have both the forward and reverse strand. As I mentioned, you need to remove the reverse strand. Cluster is formed, and then so these are different clusters. So one cluster is going to have around like 4,000 molecules. In the cluster, you have both a single forward as a reverse strand. Now you are going to remove the reverse strand. Now you are left with the forward strand, and you need to perform a sequence primers will be provided. So the forward primers have been provided, sequencing primer. This is a DNA insert which you want to sequence. All the 4 nucleotides will be introduced. Now, depending on whether they are complementary or not, you are going to bind. And at every cycle, pictures will be taken and they will be identified. So every second, one base incorporation is being identified and the sequencing is being done. So because since the sequencing is happening during the synthesis, the terminology name is given as sequencing by synthesis. So million cluster, all of them are being sequenced at the same time. With one product, this will be miniature and removed. Index 1, sequencing for that you require a primer, and index 1 sequencing is being done. This will be then removed. Index 2 sequencing. Once index 2 sequencing is done, there will be read 2 sequencing. So for peer-led sequencing, you need to generate a cluster again. So this is reclustering being done. The reverse read sequencing will now proceed. For that you require the read 2 primers. And similarly, by following the sequencing by synthesis method, read 2 sequencing is completed.
We are detecting the bases at here with the fluorofluoride. Why can't we detect when both chemical is so that
okay. Backward.
Here, I mean back. Little back. Yeah, why let's can't we detect anyway using the basis here? Why can't we detect just here? Or where we are making the complementary strands here?
This is a forward scan and this is reverse. This nucleotide and this nucleotide, they are complementary to one.
But still we are using the nucleotides from the...
Yeah, when you incorporate when you like provide the nucleotides, the one that is going to bind here and the ones they are going to bind they are going to be different. Let us say you have A, obviously it will be T, right?
Yes.
So when the nucleotides are provided, A against it will T they will bind.
Sure.
But T against it will A will do that, right? So when we try to collect the information, because this has A has a different fluorescent molecules, while the nucleotide that is going to bind here will have a different fluorescent molecules. Okay, so these fluorescent molecules they are going to mix up. Okay, so the system won't be able to identify what is the actual nucleotide that has been bound in the particular site. Okay, so then why we need to have a unique place for the fluorescent molecules also.
I got your point in this. But how the system is identifying that this time they are not using the fluorescent tags?
Every cycle, only one of the four nucleotides is going to bind. As I mentioned earlier, this 4 A, E, G, C, they are bound with different fluorescent molecules which are emitting radiation in different wavelengths. Okay. So at the end of every cycle, we are taking 4 pictures in 4 different wavelengths. Okay. So if let us say we have red, green, blue, and yellow. 4 pictures in red wavelength, green wavelength, yellow wavelength, and last one blue wavelength. Okay. So when fluorescent nucleotide molecules, let us say it is labeled with the fluorescent molecule emitting blue wavelength. Okay. When it binds there, when the system takes picture, it will be visible only on the blue wavelength. It is not going to be visible in the other wavelengths. That's how the system is able to identify which nucleotide has bound in the particular cycle.
To generate the accuracy of the capturing of the images. Different four colors are there, no? So for accuracy purpose, you have to remove something for more accuracy. You cannot have simultaneous shading both.
Sure. I got this.
You confuse the machine
The camera.
for capturing the images. Therefore, one has to be removed for better identification of the one which we want to identify.
But when you are like in the stool sample example, when you have multiple bacteria, the different things will bind at different times. So that is not because they are also the same thing that they are not uniform.
We are seeing, let us say you have a fluorescent. 10 clusters are there. At a time, all these 10, they are not going to bind a single nucleotide, same nucleotide. Every cluster, the nucleotide molecule that is going to bind will be different. So let us say you have a closed cell surface. There are 10 clusters, right? Now 10 clusters at a time, they are not going to bind the same nucleotide molecules during the sequencing. Multiple nucleotide molecules will be there. But the wavelength emitted from these nucleotide molecules can be identified through the optical images that will be taken by the engineers. And this will help you to identify which nucleotide has bound. Not clear?
Because it's the same thing, so it is taking different data at the same time.
Yeah.
Same thing here also, the different thing will bind for the forward and reverse. So we remove the reverse. So you get a pure data with only one nucleotide. When you are having multiple targets of multiple organisms or because of anyways the machine is getting mixed data. So mixed data
You are getting the entire data synthesized, everything is been synthesized. Now the question comes ki there are 50,000 bacteria are there are 1 lakh viruses are there. What would be the point? So how do people are accurately identifying ki acha, we have got in that entire genome these many base pairs belongs to this particular bacteria. Even in E. coli also
They
we have
will be
multiple
done during
shapes
the
also.
data analysis. All the data will come together as a single FASTA file from the bacteria, it will just give out the FASTQ files to you. But when you perform the downstream analysis, this FASTQ files, obviously we are going to align to some kind of database references. There at that time, you will be able to identify what are the diversity of bacteria, what is the virus that is present here. There is like after downstream analysis. But sample to sample, you will be able to segregate the data depending on the index information that you have given. I have my sample and I have your samples. If we are putting them together, obviously let us say my sample is also a mixture of different microorganisms. Yours is also a mixture of different microorganisms. But when we provide, we are going, my sample is going to have a unique barcode. Yours is going to have a unique barcode. At the end of the sequencing, FASTQ will be generated separately depending on the unique barcode. But when we perform downstream analysis, we will be able to identify the types of viruses, the types of organisms, bacteria, that is present. So this is how.
Can I just
Yes.
like he mentioned, there is a panel, respiratory management panel. So now you know the viral infections are rapidly present now because of the climate. You will have dengue, you will have influenza, other virus infections simultaneously occurring in the community. So that panel will identify, there are say 8 million type of patients related different type of likely possibilities of different viral infections. But the panel has got reference data have to identify the differences existing in the community. Even if the syndrome is same upper respiratory tract infection, but it could be caused by more than one virus. Or it could be other way. The same virus could be there and it could present like it happened in case of COVID. It presented in different clinical form. So there are these type of technical possibilities. There could be one syndrome caused by more than one microbial agent, and there could be one microbial agent which could cause more than one syndrome. All these identifications are possible with the help of this type of tools, and therefore they are getting the attention of working into our diagnostic microbiology. Molecular tools are in plenty. It depends on our wisdom to choose which one to be used for our accurate diagnosis. More accurate you are in your diagnosis, better treatment regimens you can bring. This is what I would like. You agree, sir?
Yes, sir.
Just one basic point from my side. I would like to know if anyone needs to set up any NGS lab,
Yes.
and they per se have their molecular setup for open platform, closed platform RT-PCR system. So what specifications we need to get it to have NGS with us, or whether the same RTR platform, molecular platform which inbuilt in-house it is available, can we have NGS with us?
When you have the basic molecular biology, most of your decoded users of biology, most of the minor accessories, instruments required for supporting NGS experiment, we generally actually will be there. Likewise, maybe the needing travel sectors. So this is one. Then you might be having DNA quantification machines or nucleic acid quantification machines. Additionally, we also recommend certain accessories instruments. Okay. So when we perform obviously being a molecular biology lab, there might be separate room for DNA extraction, right? Separate room for performing PCR, and separate room for another machine. So we also suggest post PCR rooms. Okay. So the first basic instrument that will be required starting from your DNA extraction and extraction, you need to quantify the DNA, right? So for that we have a machine that is also based for the tubing. We normally do not proceed with the nano drops. Okay. We have to try this one because this is a fluorescent based quantification machine that gives you accurate quantification of your DNA or RNA. Then
Sorry, I have to interrupt because if suppose I have nanodrop available for quantification, so what basic disadvantage is there for nanodrop?
Nanodrop, basic disadvantage is nanodrop can quantify based on the nitrogenous gas that is present. Okay. So even if your DNA is fragmented, or even if it is so much fragmented that different nucleotides are present, when you quantify with nanodrop is going to give you some quantity. Right? But your tubing, they use certain fluorescent probes which bind only to double-stranded DNA molecules. Okay. So because of that, it will give give more accuracy in quantifying and particular nucleic acid. So that is the difference.
As per what I had a previous knowledge, basically it gives about the purity of the extraction which you have with you.
Nanodrop gives you the purity. 260/280 ratio. We are going to utilize that as well. Plus for actual quantification, actual quantity, we utilize cubing because cubing gives you more accurate quantity. In nanodrop, if the particular concentration, let's say, is coming around 100 to 150 nanogram per mole, but that may not be the actual concentration of the particular sample. Because we need to have a good quality. Because in order to have a very good quality, as well as the quantity of the nucleic acid, which relies to it, as well as nanodrop give you the purity, 260/280, whether it is protein contamination, other organic contamination. And when you go with QA, that gives you the accurate concentration.
So are we going to discuss this part also, purity?
Yeah, we will discuss
purity
during the live preparation. We will discuss. Then once it is done, you need to have another machine known as the tape station, or we call it this actually fragment analyzer. So when you are having starting materials like RNA, we need to check the integrity of the RNA. We call it the RNA integrity number. So this fragment analyzer helps to identify whether your RNA is of a very good quality or not. Another way it will help to check the QC of their RNA molecules. Okay. So there are different processes for that. So fragment analyzer is required. If you do have this two machines and you are able your nucleic acid pass the QC, then only you will be able to proceed with the library preparations. Okay. So once library preparation is there, in the library preparation also there are minor instruments which are required. The pipettes and all this will be already available in your lab. Then we can require thermal cycler. Suppose it is a silicon-based kind of application, you need to amplify it, right? We need thermal cycler, but then once the library preparation is done, we will quantify the library and check the QC using the QB as well as the tape station or the bioanalyzer or the fragment analyzer. Then machines pass it, and once the QC pass, the library QC pass, we will then go over the
So basically additional equipments might be required for NGS.
Yes.
And if we have pre-PCR and post-PCR setup, so that is
that is fine. That is where the instrument will be placed.
Correct.
Okay. And for the NGS machine room, you need to have a specific environmental condition. The humidity should be around 50-60%, and you need to have because you are going to run machine for 24 hours. We need an optimal temperature condition for around 20 plus minus degrees centigrade.
But if that these are the specifications for other RT-PCR platform system also, so we can keep NGS and those platforms together. Would it be
That can be placed whether there is no issue. But for NGS, we require a separate table, non-vibration table. But you need to provide optimal temperature, optimal environmental condition.
So do you recommend any screening before the NGS?
Screening of what, sir?
The difficulty level which you was trying to get?
Screening of the environment, your lab.
Before the preparation of the first library, do you recommend any screening procedure so that more accuracy can be achieved?
Sir, actually we are supposed to do a layer swap from wherever we are.
That's the environment.
Yeah.
So apart from that, so suppose I was thinking whether there is a step 1 as a screening, and after achieving result of the screening 1, you proceed to the second one, like genomic sequencing part A and part more next generation sequencing part 2. More accuracy to derive. You can say ultimate result is available in NGS. Complement to that, do you recommend any procedures to achieve better accuracy?
Better accuracy in NGS data than of sequencing. In case we can discuss. Like, see, initial thing is until and unless you are not able to achieve the QC recommendations of the initial input, we are not going to proceed with the library preparation. Okay. Let us say you have a nucleic acid extracted from some kind of microbial samples. Okay. So every library preparation we have a basic recommendation, input quantity recommendation as well as an input quality recommendations. Until and unless we achieve that, that we normally do not proceed with the library preparation. Because if you are not able to achieve the concentration as well as the quality, you at the end of library preparation, you are going to get a very bad pressure. Okay. So that's why normally we determine the base extraction kit for RNA or your DNA extraction. And as I mentioned, we need to have qubit as well as the fragment analyzer or test station, which will help you to accurately quantify as well as check the QC. Okay, once the QC is done, normally proceed with that. And then sometimes it may happen during COVID, for example, we need to perform the RT-PCR of the samples. Because depending on the CT value of the samples, we proceed ahead with the preparation.
Screening.
Yeah, this is a kind of screening. So the CT value should be like below 20, like below 25, or below 30 also, but below 25, below 20 gives you more accurate result. That gives an idea that the sample is very good.
I will quote another example. Like in case of this is one aspect of molecular technology. Another thing is when we are suppose we are all, I assume that there are microbiology people around. We used to get sample which was contaminated by non-pathogens as well as commensals and pathogens. So we want to know who is the pathogen. So what we do, we decontaminate the sample by treatment with some acid or alkali, remove the unwanted organisms from these clinical samples, target the desired pathogen to be identified by some screening procedures, Yes, it could be urine sample, it could be sputum sample, it could be anything. So these are microbiological pre-treatment of the sample before it is subjected to final identification. Similarly, I am trying thoroughly whether any such procedures are in recommendation from you for proceeding towards the NGS.
Yes. So depends on the applications. We have certain regulations. Let us say you are collecting sputum samples and you want to identify organisms there. When you collect the sputum samples, obviously there will be host contamination will be there. So we have, as such, we do not provide a particular kit, but in the market there are various providers which can help you de-host. We call it de-hosting kits. This will help you remove the host genome host cells, and that's how you will have only the organisms, your microorganisms, nucleic acid molecules. So those kits, those recommendations will be there in the depending on the application you are choosing. So it's there.
I think that will be part of this.
Yeah, this is a part of the library preparation.
So I think that topic is maybe qualification and quality control of input of DNA. So that part you want to do.
This is just what we have discussed. The first thing is the library quantification and quality control. Whatever we have been discussing, we are not going to exactly do this as of experiment because initially the program was to do this. So we have like remove few steps which will like take more time. So the time. So this quantification and quality control of input DNA. As I mentioned, for quantification, I'll show you how to utilize this qubit later on at the end of the library preparation. So
That is the second half after.
They will give you an idea how DNA can be quantified.
But still, I would really request you because see, this is the basic heart of the NGS. If the sample which we have to run, if the quality control and the QC is not yet passing and we are going on, so we may land up in an adverse situation. So at least if you can give some tips of troubleshooting, how a good quality of purity of extraction should be there and what troubleshooting should be done, and when the QC has to be passed, so what are tips should be there. If you can give that will help.
I had a question about the treatment and qubit and test station part. As you rightly said, that in-house bioanalyzer test stations, these are costly. Not all places can afford to keep them. The nanodrop and the RNA gel sequencing, gel electrophoresis, that can be done in-house. Our personal experience is nanodrop will give you a good value even if the integrity is bad. So we usually pair it up with the RNA gel electrophoresis only if the band is sharp and all. We understand that whatever nanodrop value is, the integrity is also good. We have a bioanalyzer, but the kits are very short shelf life.
बायोलॉजी अभी डिस्कंटिन्यू हो गया
It's actually not even available, spare parts are not available. I wanted to understand that date station and qubit, both are must for go ahead with
in
it.
order to have a very good quality library and our recommendation is to have both units with as well as any kind of fragmented both are must.
Date station is giving the ring value. What is qubit giving it as?
It gives you the actual concentration of the I'll just show you a slide. You can please take the T. I'll also show you the slide related to this quantification and
We'll copy that.
yes. So the first thing is DNA extraction or DNA isolation or nucleic acid isolation. So at the end of nucleic acid isolation, you need to check the quantity as well as the quality of the nucleic acid. For that, as I mentioned, you will require two systems known as one is the qubit, which is from Thermo Fisher, and another it can be tape station that comes from Agilent, or it can be QSEP 1 plus that comes from Biomimetic. Okay, fragment analyzers are there. So DNA input QC check. So if you are providing a very best quality DNA, it's going to be a garbage only. So if you are putting garbage, the library that you are going to get at the end is going to be garbage only. Okay. So we need to have a very good quality DNA as well as a quantity or quantified DNA. Now the qubit. This is a machine. How the qubit looks like. So the qubit generated the quantification is based on fluorescent molecules, something like the cyber ring, which is used in your qPCR. They generally go and bind to the double-stranded DNA molecules, which means they only will quantify the intact DNA molecules. That's how they keep the very good accuracy of the quantity. In case of nanodrop, what they do is they only measure the absorbance of the nitrogenous bases. Okay, so even single nucleotide molecules, it also gives some kind of absorbance, right? And hence they do not give the accurate concentration of the DNA molecules. Even the fragmented DNA molecules also be quantified. So that is not true. Because if you are using a fragmented DNA molecules, you are not going to get anything at the end of the library preparation. So DNA should be intact, it should be properly quantified. So that's why we follow the qubit quantification method, right? Now in certain applications, these are certain ways. So 260:2 ratio, 80 ratio, this is also necessary. This can be done using your spectrophotometry and nanodrop. This is a recommendation here for RNA, that is 1.8, 260:280 ratio. For DNA, sorry, it is 1.8. For RNA, you need to have around 2.0. The 260 and 230 ratio, this gives you about the contaminations. It need to have a very good quality of good values of around 2.0 to like 2.2.
So it's not coming in this way, you should discard that?
No. It's up to the user to decide. It is a very rare samples. Obviously, sometimes it is difficult to get samples also. Sometimes we push it ahead with them. But normally we just focus on the 260:280 ratios. Nearly 1.8, if it is something below 1.7, 1.6, sometimes we go ahead. Why? Because you may not be able to collect the samples for a second time. So in that scenario, we have to consider it here. But the optimal condition, the ideal condition is the value that is given here. So contamination check done by spectral nanodrop. You can also sometimes check the integrity of DNA using a gel, right? The gel also will give you an idea whether DNA is intact or fragmented. So this can also be done.
What is electrophoresis?
Yeah, this is gel electrophoresis only. So most of you might be using this one. This one is the qubit quantification, as I mentioned. They usually use the fluorescent molecules which will bind only to the intact DNA and help you quantify the actual concentration of the nucleic acid. So, base recommendation is to quantify in replicates to actually calculate the actual concentration of your sample molecules. So this is, I think, normally most of the molecular biologists might be following. I mean, when you quantify, we normally quantify in duplicates or triplicates, right?
What should be the ideal cutoff for
Concentration? Depends on the type of application. Because we have a input recommendation for every application. Some application might be requiring around 50 nanogram, some application might be requiring 250 or 500 nanogram. So it's up to the application that you are following. Because, for example, you are having a formalin fixed paraffin embedded tissue, concentration is going to be very low. Okay, for those kind of applications, specific recommendations are there. What should be the QC value? What should be the input quantification value? For other applications like total RNA sequencing, the input RNA concentration and the RNA integrity number, as I mentioned earlier, those recommendations are there. So every library preparation or every application, they will have their own input recommendation and as well as QC recommendation. So you need to just fulfill that. And most of the time, you will be able to fulfill that. Certain challenges will be there, but most of the time we normally are able to fulfill that particular requirement.
So for sequencing, just like sequencing, this is the whole quarter paradigm, the quantification or purity. Because sometimes we extract such that the quantification is very good. It's like near about even 500 nanogram. But the purity is lower. It's lower than the cutoff of 1.8 or something, say 1.5, 1.4 like that. So you say that is a better sample, or suppose it's like just 100 nanogram per microliter, but it's like 2. Absolutely 2 is the QC. Would that be a better sample for this?
It all depends what is the value of the particular sample. So if the sample is very important to you, means you can decide accordingly. Normal recommendation, whatever I have given, is the ideal conditions. Okay, even if you are able to achieve the concentration, and if your 260 by 230 ratio is deviating a little bit, okay, instead of 1.8 you are having 1.6. If the sample is very rare for you, very important for you, you can proceed ahead. It will give you some information, but that information may not be like, you may not be able to achieve the 100% or extract 100% information, but you should be able to get certain information from that particular sample.
Is time factor very critical for every step? Time factor,
Time factor is very critical, sir.
any indication of how
So when we follow the library preparation steps, there will be certain time factors. Let us say it will indicate that you need to incubate the library at 55 degrees centigrade for 10 minutes, and it should be actually 10 minutes. For example, during fragmentation, when we fragment the DNA, if you are fragmented, the enzymatic fragmentation will happen, right? So the condition will be provided for incubation. Let us say 35 degrees centigrade for 10 minutes, let it be 35 for 10 minutes. If you go beyond that, their fragment will become smaller. If you go less than 10 minutes, your fragment is going to be larger. So they will impact the quality as well as the quantity of the library and there. So those things are very important. So this is the library molecules as I mentioned earlier.
How stable? We know that RNA is very unstable, DNA is very stable, and what is the status of cDNA? How stable or unstable it is in comparison?
If you convert the RNA to cDNA, it becomes more stable.
But is it lesser stable than a double-stranded DNA or like?
Now it is also a double-stranded DNA.
But at least the first strand is still in.
Now you need to convert it to double-stranded DNA molecules, not just the first step. Second step also will be converted, and you have the double-stranded DNA molecule, and then you can store it for some time. Because RNA, for long-term storage, RNA may get more degraded, right? If you have the RNA molecule, convert it to cDNA molecules, you may be able to store it for some time. So these are some of the fragment analyzer which was available, which is available in the market. So tape station is the currently most of the labs are using this. Besides this, we also have another machine known as Byramjee Q7 Plus, which is also a kind of fragment analyzer. Now, during fragment analysis, it's going to give you an idea of the size of the library. At the end of library preparation, we are going to run the libraries over this tape station, and it will give you an idea of the size of the library. Okay, so the same machine can also be used for checking the QC of the RNA molecules, because they have different kits to do that. Okay, the QC of the RNA molecule will check by identifying the RNA integrity number. Okay. This is the 2%. This is an environmental condition that I was mentioning. So ideally, a lab should have a temperature around 20 plus minus 2 degrees centigrade. Humidity normally we recommend around 60%. This is elevation. Sometimes when you have a altitude is very high, that time it may affect the run. So in place like Chennai, Mumbai, Pune, so depending on the area, the humidity, the temperature as well as elevation is going to differ. So we need to provide the optimal environmental conditions. So in order to do that, we recommend using dehumidifier, which will absorb the excess humidity, and you are going to have obviously ACs will be there, centralized ACs will be there to control the temperature. Okay.
There is something called Q-score. Can you elaborate something on that?
Q-score. This one. This is the quality score. So every NGS flow cells, there are specific quality scores recommendations from every flow cell type. When you perform your sequencing, at the end of sequencing, there are certain quality scores will be generated. So when we say Q30, 85% Q30. So when we say Q30, it gives you an idea, means it tells you that for 1000 base sequence, there is chances for one nucleotide in an error. Okay, that is mean by Q30. 1000 bases sequencing of 1000 bases, one error in 1000 bases. Okay, when I say 85% of bases is higher than Q30, the machine particular sequencing flow cell is going to generate data, and all those data, 85% of the data is going to achieve the Q30 values. You get an idea of that, right? So data is going to be very much accurate, 99.9% accuracy. One error in 1000 bases is like 99.9% accuracy. So every flow cell they have a defined Q30 values. Here if you are performing, let us say 150 bases to close 150 bases. The Q30 values that should be generated should be like 80%. This gives you an idea that 80% of the data being generated, they are achieving 99.9% accuracy. So chances of error is very clear.
So what you were mentioning at that time, let's say we are sequencing the whole genome of a human cell. So you are saying 30 times we need to run the, so that 30 does not have any value, so any significance in this 30?
this is different. 30 times means you are sequencing for the one time, then the second, third, 30. Entire genome is sequenced for 30 times.
That is different.
This is different. This gives you an idea of the quality of your sequencing machines. Q30 here means when you are sequencing 1000 bases, 1000 nucleotides, there is a chances of one nucleotide being an error. Okay, gives you a 99.9% accuracy here. 85% here means if you are sequencing, say every flow cells, they have a recommended Q30 values. So one flow cell might be having there will be a specification that this particular flow cell will generate 120 GB of data, and out of that 120 GB, 90% should achieve Q30. Okay, which means out of this 120 GB of data, 90% of the data is going to be around roughly less than 100 GB. Right? 100 GB will have 99.9% accuracy.
And then one stands for forward primer and two stands for forward and reverse primer.
Yes.
And 75 base pairs or how many base pairs we go to talk about?
Yes. I think we can start with the library preparation, and simultaneously, like when we go for in every step, we can discuss all these things. So you must be having the protocol with you. Chapter 2, library prep. Then you can see here introduction, extract RNA, anneal RNA, synthesize or strand cDNA, amplify cDNA, segment PCR amplicon, post segmentation cleanup. So these are the actual workflow of the library preparation. Okay. Now, because it can be any kind of virus. Okay. It can be any kind of virus. This particular library preparation protocol can be used for other viruses as well. Okay. Right now we are going to prepare libraries of COVID. Okay. For that, primers are provided which will amplify the COVID viruses. Now, if you want to sequence other viruses, you need to just change the primers. Okay. You need to just change the primer. You need to design the primers and just change the primers. Here, in this example, we are going to use primers which will amplify the COVID viruses.
The oligonucleotides on the chip and the adapter sequence.
They are universal. They are universal. Okay. The first thing is extract RNA. RNA will already be extracted because you might be collecting in samples during COVID. We have collected in this particular tube. I forgot the name. VTM, sorry, VTM. Okay. So VTM, you have collected the samples. You extracted the RNA, right? So then those are extracted RNA. You go for checking whether that sample was positive or negative using the real-time PCR. At the end of real-time PCR, you get an idea of the CD values. Then this is a kind of QC check of the particular sample. So normally, we recommend, as per our experience, QC values which is less than 25, they give a very good library. Okay. So normally, we go for samples having a QC values of less than 25 or around 25. Okay. Even though you can get libraries out of CD values having around 28, 29, but what will happen when we assemble the entire genome structure? There might be some pieces which might be missing out. Because those kind of samples, sometimes what happens, the sample viruses load are very low. That some fragments might not be amplified during the PCR protocol. Okay. So CT values around 25 to 28 is fine. This can also be done for other viruses. You need to know the CT values. If it is very low CT values, then obviously the load of the virus will be very high. And from there, you have the RNA with you. You need to design the primers for specific influenza virus, dengue virus, if you want to sequence those viruses. Okay. So,
what if the value is CT value is more than 25, 30? How do you deal with the sample like that?
Sir, when you obviously you know that when you have a lower CT value, the concentration of virus is very high, right?
Yes.
you need to optimize that. Means sometimes maybe CT values around 29, 30 might also work. But when you try to assemble the library for your sequence together, you might be missing some parts of the genome. You may not be getting the entire genome.
How do you reduce it to below 25?
Yes, sir.
How do you
He's
reduce it
saying
to
how
below
to
25?
concentrate that, like it's a diluted thing that
Can you dilute the sample to bring it below 25?
Is there a way to
No, CT values, I think
It will be related to the
higher the CT values, the more the sample is diluted.
virus load is
Viral load is very much less when your CT values is very high.
okay.
When you have a lower CT value, the virus load is more.
Yeah, you wanted
We need more load of viruses. We need like
so you can concentrate. You can
high
concentrate and then bring it below 25.
But from a swab, how can you further concentrate the sample? If it's a bronchoalveolar lavage or something, you can centrifuge and depose it directly. But from a swab, like in case of COVID samples, how to concentrate the sample?
How do you concentrate?
Ultracentrifugation, can you recommend ultracentrifugation of the sample which is having higher CT value so that the virus is more concentrated and that sample is then suitable? There are affiliated centrifuges which allow the viruses to sediment and that will give a
Okay.
Likewise, I got
Okay, sir. I got the point.
What one?
Yeah, if you are going to give a pure culture of an organism, that is fine. That is fine.
There is a situation, a patient is on ventilator and we are suspecting a COVID diagnosis. In that sample, the sample becomes very rare and crucial. It's an aspirate from the tubing, which are one for the ventilator. On that sample may be having lesser quantum of the virus. We want to concentrate. In that case, we will recommend the refracted centrifugation to concentrate the quantum of the virus, make it suitable for the analysis, and then use it for the sample for the preparation. Likewise, that.
See, what
That can also be done.
I also have a question in this line. You listen to the question then because your answer will be common for both the questions. So you see there are not always stool-like samples will not be there from human body. Stool is absolutely so rich in microbes. Okay. There will be possibacillary samples also. Okay, where the pathogen load is low. So as sir's question is that the patient is infected with the COVID virus, but the load of that virus is very low, so that your CT value is like something higher than that is 30. Okay, threshold is higher than 30. So how to bring it down? In case of my question, suppose we have a possibacillary sample, the pathogen is still there, it's still a pathogen. Only the number of nucleic acid count for that pathogen will be lower in that sample. Then how do you get above this? This is the obstacle that we have here, the CT cutoff of 30 or something. Then how do we get above that and go ahead with the rest of the process?
So it actually depends on the kind of question you are asking, your research question you are asking. So let us say even if you are having a CT value about 30, around 30 also, you will be able to identify the virus. The only difference it's going to make is you may not be able to assemble the entire genome of the virus, but you will be able to identify the variants or the types of virus that has been infected. Okay, now if you want to get the entire genome of the virus, in that case, higher the load of the virus, you will be able to assemble all the genomic information. That is the only difference I have was.
We want to do HPV genotyping and tract resistance testing for some patients where we could not able to proceed for sequencing since the patient is having a very low viral load. So we took up like a small kind of project, and it's not actually a recommended procedure, but still we try to do some concentration in the extraction procedure. Why I will take multiple lysate. I'll take the lysate buffer, for example, 2-3 volume, and I will add the recommended sample volume. But I will just transfer all the lysate to the same column. We do column-based extraction method. So when I pull all that area of viral load is somewhat increasing, and we have achieved that procedure for one patient where we can achieve the sequence. So maybe, but there is no literature.
Another application that you can do is, if you remember that I have shown the RUP respiratory virus oligopanels. Okay, so there are multiple such kind of panels which will help you the types of the virus, type of their genotyping and everything. So even if you do not, even if you have a virus concentration which is very low, those kind of panels will help you identify the virus type as well as the variant type also. Because these are like a kind of a surveillance panels which will help you identify where the entire intact genome is not required. There may be one solution. But when you need to build a COVID genome sequencing, this is like the library preparation protocol when we initially introduced to develop the entire whole genome of the virus. When something which is let's say very much novel to you. Okay. So that time this was introduced. But we also have different panels, as I mentioned, virus panels, which can be helpful in identifying the virus, the genotypes, their variant types. In this kind of panels, what happens? Only those genes which are important, they are just amplified and they are just sequenced. So those can be one solution for you. Or another solution is, as I mentioned,
Different samples from same patient.
Yeah, different samples you can pull them together and collect the acid and you can proceed.
So one more related question to this. Can we do it, can we sequence it directly from the sample by after extraction of the nucleic acid? Not
We
doing PCR.
normally-
See, what you said is you do the PCR, you detect the virus and the CT value.
We can also do that. There is in some scenario you may be like the particular samples may be positive. There is you are having symptoms of that particular
Yes.
organism. Okay. And let us say you don't have an instrument like real-time machine to perform and identify, calculate the CT values. Okay. In that kind of scenarios, the same type of panel that I was mentioning, that can be used because we have different virus panels. 3,000 virus panels, 4,500 virus panels are there. Okay, that can be utilized to identify the virus of the infection.
Let's say I'm suspecting such a kind of virus which is not integrated in your viral panel, RUP. Then how can we still proceed with that?
You can still proceed, but in that scenario, that may not be the optimal way, but you can still proceed there. In this scenario, what will happen? You may need to give a higher depth of sequencing to particular samples that you have. Let us say higher depth means like if the panel, if the virus is not a part of that particular panel. Okay, there are two ways. Okay, if the virus is not a part of the panel, what you can do is obviously there might be some literature information about the genome, right? Design the primers, okay? Amplify with your the samples with these primers. You certainly will get some amplicons if it is positive, right? From that amplicon, we can proceed ahead with the library preparation. That is how we are going to do here with a similar way that we are going to proceed with it. Or another thing is there is a short-term metagenomics approach. Okay, you take the samples, you prepare library directly out of the sample. But there you need to provide higher depth because there might be some kind of cross-contamination from the human sample. Let us say human genomic DNA might be coming up. Okay, so you need to get more data out of that particular sample. And when you align with the reference genome, you will be able to identify the particular virus. These are two different approaches.
Since you mentioned the short-put sequencing also, so I think nanopore sequencing is one of the. Now, how do you compare that advantages, disadvantages with your advantages?
Nanopore, there are low fragment sequencers, and their data accuracy compared to Illumina will give around 99.9% data accuracy. And nanopore's accuracy may be around 60 to 70%. So there is a difference. When you try to identify something new, you need to have a very good accurate data. Yeah, this is the best source. So let us go through the protocol. So once you extract your RNA, okay, so depending on your application file, you can choose your real-time PCR to have the QC values, or you can even choose the, as I mentioned earlier, you may utilize the tape station to check the integrity of your RNA. Okay, but for this protocol, once your for the COVID protocol, once you extract the RNA, we normally follow the RT-PCR and choose the base samples, okay, which are having CT values, lower CT values. Now, the extracted RNA you need to convert into cDNA, okay. So, for that we use some random primers which will bind to the RNA and then will convert the cDNA, the first strand cDNA and the second strand cDNA. Okay, the cDNA molecules will be converted, will be then later on amplified. Okay, so here for amplification we are going to provide the primers, right? The primers will then amplify the target molecules, which is going to be your viral genome. See, because when you extract the RNA, obviously some human RNA will also be going to be there. When you convert the RNA to DNA, obviously some human DNA will also come. But when you amplify with the primers, virus primers, at the end of amplification, at the end of your thermal cycling step, what will happen? You will get only the virus amplicons, right? Because your target, only your virus target will be amplified here. So you have the virus amplicons. Next step is to go ahead with the segmentation process, which is cutting down your nucleic acid for the virus amplicons.
Sorry, I have one question here. So here in step 2, again I'm having to the primers.
No primers required. Means whatever samples, let us say something, even when the COVID was initially COVID started, okay, the samples they go for the de novo type of sequencing or the shorter metagenomics approach. Whatever the sample extract nucleic acid, okay, go for segmentation. The library preparation is done, right? But you need to give a higher data, so that you will be able to assemble and form the genome of that particular organism. So the approach will be different. Here, when we design a primer, these are for those viruses which genetic materials is already available in the database. For example, like dengue, influenza viruses, monkeypox viruses, those genetic information are available. You design a primer for that, amplify the viral genome, you get the amplicons, you proceed ahead with the library preparation.
But when the unknown disease, you don't know if it is a virus or bacteria or fungus or whatever. So do you give a mixture of all primers there or no primers and that step is totally bypassed?
There is no primer. See, when it is totally new, there is no primer that will be available, right? So there is totally different protocol. We call it shortcut metagenomics approach. There is totally different protocol. Okay. So once you have the cDNA, you are going to undergo amplification of the cDNA using the primers for the specific viruses. Once your amplicon is ready, you are going to proceed ahead with the segmentation, where your DNA fragments will be cut down by specific enzymes. And after segmentation, what will happen? There will be some post-segmentation cleanup. Okay. After cleanup, there will be amplification of this segmentation products. During this amplification, what will happen? The adapter molecules or the index molecules, they will be ligated, okay, to the ligand molecules. The adapters, they are going to be ligated, right? So once the adapter is ligated, at the end of this application, you are going to get the complete library molecules. Now these complete library molecules, you need to purify them, obviously. Why? Because during this ligation, two adapters molecules might be ligating to one another as well. So you need to purify them. So we have a cleanup process there. That once the final libraries are made, we will quantify them and we will check the QC of the library molecules by using a fragment analyzer. So this is how the workflow proceeds. Now in order to save time, what we have done so far is like the RNA molecules was already with them because these are COVID samples. They have extracted the RNA. and they have converted the RNA molecules into the cDNA molecules. So today we are going to start with the cDNA molecules. cDNA molecules is already there, and they have also amplified the cDNA molecules. Because this amplification step normally takes around 3 hours. Okay, in order to save the time, we have completed up to that particular step. So today we are going to proceed with the segmentation steps. Okay, where your amplicons will be fragmented, and afterwards adapter molecules will be added there. Okay.
We were 170 yes. 15 ml found. What is PCR there? So, what are the consumables?
EVLDs.
Awareness with the most.
So we kept it outside. Sorry. Because now we have to set up this thermal slider, 55 degrees, 5 minutes, overhead, and 10 Celsius. Anyone of you can do that?
Somebody will have to do it, no? Please come.
He is an MD in microbiology.
Yeah, that's better. We will learn through your experience.
Say that. Say that.
Can you please mute the mic so that everybody is able to listen to you?
Yes, sir.
Please give him a mic.
I will program it. Just check.
55 or 5 minutes. What is that?
Play. High safety level.
Hide it.
Mic down there.
Yeah.
Yeah. Please repeat the instructions so that everybody is able to be with you.
Okay, so here we are doing fragmentation in thermal sample. So the condition is 55 degrees Celsius for 5 minutes. So we'll keep the samples in there.
Tag program. Sorry.
Why is it called fragmentation, tagging plus fragmentation?
Yes.
Combined protocol.
Tagging and fragmentation, again taking this developer.
Tagging and fragmentation.
Okay.
Volume is 50 microliters.
Volume size is 50 microliters.
50 microliters.
Yeah.
So,
How will you load the sample? Loading?
No, just we are
Setting the machine. Setting the machine
Okay.
first.
It is 55 degrees
Okay,
Celsius
what
for
is the
5
next
minutes.
step? Level you
10 degree infinity.
10
TAG1.
degree for infinity. Then we have that shall I put it
Yes, please level this one new 96 paper TAG1. TAG1. Now on page number 10. Please come. So before transferring the master mix, we have to transferring this code 1 and 4 to place out. Before that we are making master mix. So in 15 ml alcohol, please take it out. It's less, so we can make it multiple pixels is there.
What are the things you are adding for the master mix preparation? And
So we, as I have discussed earlier, these two, the TBLTs and TB1, this one. Page number 9 itself. Now page number 10, we are going for preparing master mix. Yeah, so point number 3. So I think this is for 96. So we have how many samples? 10. So prepare accordingly. Calculate it.
यहाँ पे TB1 और TB80 लिखा है। एक सैंपल के लिए 12
make it for at least 11.
हाँ, एक सैंपल के लिए 12 माइक्रोलीटर और EB80 4 माइक्रोलीटर। तो हमें मल्टीप्लाई करना होगा इसको मतलब 10 से करेंगे, 10 से करेंगे।
एक पिपेटिंग एरर
नहीं, वो पिपेटिंग एरर करके ही दिया है।
So all is in
हाँ, तो हमें
12 into
तो 10 से मल्टीप्लाई करेंगे तो 120 और 40
make
माइक्रोलीटर
it
होगा।
note somewhere.
तो कौन बनाएगा?
Come later. You make it this master mix. Yeah, everything is there. You can take it to clouds and what? So in the preparation, you see page number 9 in the preparation, there is point number 1 in table. It is written that consumables name and what are the storage conditions for this particular consumables. And before using, what are the instructions you have to follow for EPLT? This one is not yes. That yellow cap one. So you have to bring it room temperature, protect thoroughly before use, and the tip for TB1 it is stored at -20, and we have to remove from -20 and throw it again room temperature, and vortex it before using. So please vortex both the tubes. If it is something specific, they will return. You can vortex 5 times, 2 seconds, likewise. Generally, you can say 10 to 15 seconds is enough. So now TB1. Take it out. TB1 buffer. Okay. Pipe it out. 120 microliter.
Yeah, it is set.
Yeah, it is set. No double press, only single press. If you double press, you take more volume. Okay, double press again. That one. Okay, now you have to pipe it out EPLTS 40. So you should keep always in this ice clear for example because it is enzyme.
So I have to mix it.
Yes, we have to mix it. Now it's written anything there. Now we have to add something. What is?
Nucleus water.
How much?
20 to 100
Yes. It is 200.
How to get nucleus?
Yes.
How to get nucleus free water?
That's commercially
Commercially
available.
available.
And you can just put and it's not important.
Nucleus-free water should be always very important. Otherwise, it will reflect your sequencing run. Then you will get to know after so many runs, yes, something is coming from somewhere.
Here will get amplified.
It is again and again increasing.
Amplified हो जाएगा.
Okay, vertex it properly. So,
40 microliters in this one.
now go for the point number 2, page number 9. Now page number 10, transfer 10 micrometer from each well of COVID. Give this plate. Where is the original plate? This is amplified cDNA from the different primer pool. OEC1 and OEC2, both primer pool that are amplified separately. Now we are going to cool it in a single tube. So mainly will assist you how to cool. So every 10 micrometer we have to cool in new plate. Here is this one. Come, first one to perform. Yeah, we can do. You can also come. Two people transfer one sample to another.
Directly on the
Beats, what are beats
Suppose we are limiting the
Almost near the beats
you're leaving, so it falls down. How healthy will
So where are our libraries now?
now it's
So we have it 5 minute incubation.
5 minutes.
15 minutes incubation.
2 minutes. 2 minutes.
इसके बाद फिर वेट करेंगे ना.
हाँ. They are all standard libraries.
If you include in 20 samples, then we should actually जिसके हम कार्टेज कार्टेज दिखा सकते incubating for 2 minutes. The calculation continuity of test page. Say the following steps to make the procedure work. So next, these steps are not there. Different. Okay. Then quantify and normalize libraries.
So, as of now, you have seen how to make libraries, how to proceed with the NGS experiments. So, here in my talk, I would be focusing on what you can do with NGS. What are the applications that you can probably exploit with the NGS? So, please bear with me for next 40-45 minutes. I'm struggling with my progressive lenses, so I'm not sure when to wear it and when to remove it. So, please bear with that. So starting with a very interesting case. So it is a story, and I would be sharing a lot of stories because story is something that remains with us forever. So it's a very beautiful story of 2014. So a boy, age 14, was having some brain-inflaming encephalitis reported to the hospital in the US, and then the doctors actually did certain tests and they found nothing. Then looking at the medical history, they found this kid was suffering with the SCID, which is a autoimmune disease. So they decided that it could be immune response. The swelling is because of that. So generally, since you all are clinicians, you know that corticosteroid is one of the things that doctor proceed. So they proceed with that. In next 1 week, the situation got really very bad. So there was a buildup of fluid in the brain. There were seizures which this poor boy suffered with. So at this point of time, one of the research group in UCLA decided to come forward and exploit this case with the NGS. They took the CSF and they decided to go for the shotgun sequencing. The shotgun sequencing is nothing, it is sequence everything, whatsoever is there. So interestingly, they did a sequencing on their mycene platform and they found that out of 3 million reads which they sequenced for this particular sample, 429 reads were coming from organism which was leptospira. And now they were having a culprit organism in their hand. They treated the boy for next 2 weeks. This boy was in ICU, came out, and the life was saved. And this is what NGS can do for you. And since you have been practicing, you know in and out of the field. So this is where probably we can lay a foundation of NGS. I share another very interesting case. It is a case coming from Mumbai, because sometimes we say that PCR is much better than NGS. Where to place PCR and where to place NGS. So it is a case which is coming from a very renowned hospital in Mumbai. They used very renowned, I would say, a multiplex PCR test for Neisseria. The clinician actually diagnosed that it should not be Neisseria meningitis. And when they actually tested it on the PCR, they found that it was Neisseria meningitis. They were of opinion that this is not right. When they went for the NGS, they found it was not Neisseria meningitis, but it was Neisseria brasiliensis. It was a completely different organism. So just imagine, a misidentification can lead you to some repercussion, even to loss of life. So a correct identification becomes really very necessary. And here is what NGS can do for you. I'm not saying that, leave your culture, leave your PCR test, but it is a complementing technology that can give you a better answers. So it is, I'm sorry I didn't introduce myself. I thought, let's introduce NGS first and then I'll go. So I'm Ankit Jain. I take care of the infectious disease, public health and microbiology portfolio for Illumina for entire India. I have been in organization for past 3 years and trying to touch base with all of you so that we can promote NGS together and save some more lives. My presentation would be spanning around our portfolio and looking at different scenarios when we are dealing with single pathogen and when we are looking at the multiple pathogens and sometimes even a discovery phase. Not to mention, these are the global challenges that we are seeing. There are a lot many outbreaks which are taking place, and then the antimicrobial resistance is actually a very big problem. I was just reading a report wherein, it is going to cost us somewhere around $10,000 trillion by 2050. Apart from economy, the loss of life is the biggest thing, and they are saying by 2050 we can expect like 7 deaths out of 10 would be because of AMR. So the statistics is very, very horrible, and we are seeing that there is continuous awareness, AMR, antimicrobial stewardship program which are also going on. And the last not the least is the diagnosis. Diagnosis has been a challenging area so far. These are the statistics, and we all are aware of it. Like more than 700 million deaths occurred, not deaths, but the cases of COVID-19, 400K deaths associated with the influenza virus, and you can see everything, including the economy losses which happened. And we are just giving the figures related to US. We are not focusing on the global. The global would be much more higher than that. And what Illumina, especially NGS platform company, is doing is they are enabling you to identify pathogens, look into the AMR profiles of these, help you to look at even the host responses and host genetics, and what are the immune responses associated with it, allow you to come up with a better diagnostic test. And not the last, but it's very important, the drug and vaccine development, because sometimes it is based on the genome information that you develop. So our portfolio, by now I'm sure you have heard so many these things, because we have been talking about the library preparation, we have been talking about sequencing. So this is our entire portfolio, which is compilation of the library preparation kits, and then certain specific kits focusing on specific pathogens, kits focusing on the multiple pathogens, what are their functions, how it can support in your area of research. I will be taking you through all this in next 40 minutes. So our focus has been, because a very, very common question that we have seen is, it is very easy to generate data. It is very difficult to analyze it. People generate data so easily, but then what's like assembling the entire genome, looking at the variations, all these becomes the bottleneck. And sometimes this is the roadblock by which, because clinician is very occupied, our system is already burdened with so much these outbreaks and all. A lot of patients are coming to the clinics. We need to address those things. So how about having something that gives you report as soon as you do the experiment. So especially looking into our infectious disease portfolio, we have developed the solution that gives you end-to-end solution. And when I say end-to-end, starting from the library preparation to the bioinformatic analysis, you don't need a skilled bioinformatician to run these experiments, as in to analyze this data. Looking at, COVID was there, a lot of people were doing COVID sequencing. But do you really think that it was so hard to analyze the data? Over the course of time, we brought certain tools that really made the life easy. On some clicks, you can actually analyze, assemble the entire genome of the virus, look at the variations which are available. All these things were available with a click of mouse. And these are the methods which generally we use in all our investigations. Be it like whole genome sequencing of the isolates, because we grow isolates in lab and then we go for sequencing in order to identify are there any changes. There are certain amplicon-based method wherein we amplify and then look into the possible mutations, possible changes that are taking place. Then this is another approach, hybrid capture enrichment. So hybrid capture is, you develop certain probes. Those probes go into the total DNA, pull out the specific regions that you are looking for, and then you report it back. And the last, not the least, and this is again very important, it is metagenomics or the metatranscriptomics approach, which is hypothesis free. You don't know anything about what is happening there, and you want to know. let's take the example of sewage sample. In sewage sample, we don't know what are the viruses, what are the bacteria which are circulating. So, metagenomic approach is a hypothesis-free solution. You can actually look at what all pathogens are circulating here. Metatranscriptomics, you look at only the transcriptomic side, which is the functional side. So, metatranscriptomic word will be hearing a lot when people are talking about the gut microbiomes. So, nowadays people are very much interested in the gut microbiomes, and they want to look at the functional diversity of all the microbes which are actually active in the gut. So, they use this approach, metatranscriptomics. So, here is, I am focusing on single pathogen, and I know what it is. I really want to see. I'll share some use cases. This is a use case which is coming from the side of food. So USFDA is the country has been importing a lot of food material from here and there, and there is a large number of outbreaks that they are seeing, majorly towards Salmonella, Listeria, Vibrio. So they are seeing all such kind of outbreaks. What they are doing is, because in US the cost of treatment is quite expensive, as soon as the food comes to their port, they started analyzing that food. They use certain enrichment-based methods by which they are growing the cultures, looking at whether it is Salmonella or something else, and they are going for the sequencing of the sequence. As soon as they sequence, they get to know, it's the culprit organism which is present. I give a very bad example. There is a, by talking to the USFDA, we learned that Maggi, which we supply to US, it contains almost 30% residue of the insects, unfortunately. So very alarming figures which are coming out. And the shrimps that we are sending them, at least 10 to 20% of the material are being rejected because it is having the Salmonella presence of Indian origin. And this is what is the power of NGS. Another use case. So duodenoscope. I'm sure you have seen this more than I know about it. So here, it was carbapenem resistant Klebsiella, which they saw, some outbreak happening. When they used the routine primers to see whether it is the Klebsiella or not, the primers were getting failed repeatedly. They decided to go for the whole genome sequencing. What they found was there was a mutation. Now it was the time to revise the primers first, because they would be missing some of those cases. So better to come up with the new primer steps. So this is what NGS helped them with. The another important thing, what happened, it is not all about primers, but it was about changing the SOPs associated with the reuse or the cleaning of the devices that came into existence. This is, again, in my one of the slides I showed the better diagnosis. It is not only better diagnosis, but a changed SOP, which is associated with the cleaning and the use of device that came into the picture. And this is a kit that we have been discussing. It is Illumina DNA Prep Kit. Really work with a range of samples starting from 1 nanogram to 500 nanograms. You can prepare the library and sequence it. People interested in 16S or whole genome sequencing or metagenomic sequencing can probably use this particular library. So I talked about the whole genome sequencing as one approach. The another approach is amplicon-based sequencing. So what exactly is the amplicon-based sequencing? So you amplify a certain region of the genome or maybe entire region of the genome, and then you go for the sequencing. So this is the approach, right? And advantage, disadvantage, I'll come on to that. Disadvantage, specifically. So if there exists any mutation, because in my previous slide I talked about if mutation is taking place, we'll lose that fragment. Again, here if the mutation is going to take place, we'll lose that fragment. But the positive side is if there is no mutation, you will continue to get the data that you are looking for. So there are certain kits available with us, like the GenoScreen Deplex TB kit. If you are focusing on the tuberculosis side, looking at the MDR cases, this is a fantastic solution. COVID-Seq is the another kit used for the, as the name suggests, look at the entire genome of COVID, and then these are the IMF flu and the IMF kit. I will be going in details, showcasing you. I'll skip this slide, and this is the TB. So, general approach is you go for the various testing, and I think if I'm not wrong, it generally takes 40 to 45 days to have the preliminary report for the TB. Am I correct?
You're right.
So 40 to 45 days. So after discussing with some of the people, I realized that things will not change. Patient can wait for 40-45 days. Is this statement correct? I want to take your feedback.
There are liquid cultures which are faster than 45 days and you can get within 2 weeks to 3 weeks results. But there are some better methods also now.
This is microvector TB you are talking about. This will require 6 weeks of incubation. Only then after that we will start the culture.
Right. So, now another question. क्या उन 16 days में और maybe 40-45 days में कुछ change आ जाएगा इस mycobacterium के अंदर? Are there chances that the bacterium will start getting resistant if we administer the wrong treatment? That possible?
It multiplies there are two groups. One is the atypical ones which multiply very fast, rapid growers. So even on culture within a week or so, in 2 weeks if we grow, there is adequate number of pathogens. For the mycobacterial TB,
It's very slow.
multiplies very slowly.
Very slowly.
Any change which comes takes time.
It takes time.
And it also to revert back also it takes very long.
So time is associated with it. It is a slow grower. But of course, but do you see that if we diagnose it as soon as possible, it will add values?
There are, in the national program, there are different modalities now. We can detecting in less than 2 hours.
Less than 2 hours. Yeah. So those are the nucleic acid based tests. The NAT test.
But the liquid cultures they usually grow a bit faster.
Yes.
This case is for the solid.
Right. 16 hours you mentioned.
Yeah.
Right. So 1 day, 2 day. Will it give you the resistance profile as well?
Yes,
To certain extent it will give.
it will give.
Rifamycin it always show. How about having something which is culture free? Now I'll not go against the PCR, but I'll go along with PCR. So you have the PCR report available with you, the NAT test report available with you. You couple it with this particular test. What will happen? Of course, that is going to show you that it is an MDR case. Now couple it with this one. It is having what it can do for you. It will do the genotyping as the polymotyping of the antibody. That is one. Then it will allow you to even look at the non-tubercle mycobacteria, which is like 100 in numbers. It is also going to focus on that. And the last but not the least is it will allow you to look at the prediction of resistance to 15 anti-TB drugs. And here are these drugs, first line and the second line. It is a completely culture-free approach. Use the sputum, isolate the DNA, and process. So with our even the new students coming in, you can achieve the result within 24 hours. So once you have the NAT report available with you, couple it with this one, and this will help you. So I would say, and the most important thing is, I'm not sure whether I haven't put it along here. As soon as you do the sequencing, you can put the data in the app. It will generate a report for you. It will hardly take 3 minutes. So overall, in 24 hours, you can have the report on your table. And this report is really going to help you to decide which drug to administer. So I believe to a certain extent, it will bring the precision in the treatment. The next one is COVID-seq. So interestingly, BGMC has been one of the big user of the COVID-seq assay. When COVID was there, people were very. This is WHO recommended. It is not FDA. As of now, in this space, if you really look at the sequencing, so the things are not IVD approved so far. Nothing is IVD approved. This is something research is only, but people have been implementing it in their setup. For example, Hinduja extensively used it. Now some of, in fact, NIRT also implemented it. There are different people. RMRC, Bhubaneswar is using it. So there are people who are using it. It was part of the seek and treat trial, and on the basis of which the WHO has recommended it as one of the widest panel available that is giving you prediction for the 15 entity vectors so far. Going back onto the COVID, is though I understand the COVID is gone story now, people are having very limited interest, but yes, still we keep on seeing certain cases. But this could be very interesting for you. The kit, the basic of the kit is the ASCO2 genome is there. The primer, tiling primers has been designed across the genome. Now you use your primer, amplify the whole genome. Go for the library preparation, which you just saw how the library can be prepared. Once the library is prepared, go for the sequencing as the next step. And once the data is generated, again put it in the app. So my friend Manjunath will be showing you how to do the analysis. So just do the analysis, and you will get to know the entire genome, the variation that are there. Okay, so now I share another interesting thing. Lot of VRDLs were established, right? These VRDL started doing a lot of sequencing. So a lot of COVID-seq assays were purchased. Taxpayers' money actually went into funnel. Now, at certain point of time, COVID was gone. So all those kits were sitting idle. What to do with those kits? So our people devised a very unique method. So as one of the content of COVID-seq is Arctic primers. You know, the primers spanning across the genome. They decided to get rid of those primers, put their own primers there. Let's do dengue. Design the primer across the genome, put it in there, and do the sequencing, do the analysis. Instead of COVID reference genome, put the dengue reference genome. This is what they did. And they started sequencing a lot of other viruses. When I say a lot of other viruses, I give you some example. National Institute of Virology, they did influenza, they did RSV, they did chikungunya. There was an outbreak of lumpy skin disease virus. So even in lumpy skin disease, which is a genome of like more than 150 kb, again, the same kit was used. They designed their own primer across the genome, and then they simply sequenced it and assembled the entire genome. And they reported that what it was and how it was different from the previous outbreak of lumpy skin. So all these things you can do. So if you are having a focus towards even certain region of your bacteria, you can design those primers, you can amplify those, you can go for the sequencing and get to know if that region is having any modification or the mutation or not. If it is having mutation, it could be region of resistance that you are seeing. Probably this could be associated. So these kind of studies can be done. And this is IMAP. This is nothing. It is just you get rid of the COVID primers and you bring your own primers. So this kit is something giving you a backbone of COVID. You can design your own things. For example, in case of PGI, Chandigarh, one of the scientists is very much interested in looking at certain regions of Trichomonas and the another one is Plasmodium. They are seeing some resistance during their treatment design, and they are focusing on now certain genes. So they have designed the primers across those genes, and now they are sequencing using this particular kit. And this is how, in large number of samples, they will come to a conclusion that whether this is the region which is imparting resistance or not. Any questions so far? Shall I go ahead? I'm a flu. So flu being like very common, and we are seeing a lot of outbreak across the world. So we decided that let's bring the influenza A and B virus primers together, and we developed this particular kit. It is nothing. It is again IMAP having the primers of influenza A and B. And these are some of the use cases, like in case of monkeypox, RSV, skin disease, dengue, people have used it. And you can actually try to validate it for your own application. Now, coming on to what if I am having multiple pathogens available. So, this is a hybrid capture approach. So, amplicon and the hybrid capture, when we use this word, the purpose of using it is that because ultimately, if we really look at the total DNA would be having everything in it. You cannot sequence everything. So we need to focus on specific region. So that's why you can design your primers. And here in this case, it would be your probes which are going to hybridize to your DNA, and you can fetch it up. And the good thing with the kit, with this approach, is say for example, in case of amplicon-based sequencing, we learned that if there is any mutation, there is a possibility that the primer will fail. The amplification would not be there. Here, it is going to tolerate a lot of mismatches. Even it is not finding the exact fragment. To a certain extent, if it is there, it is going to still bind, and it will give you the sequence. And once you have the right sequence available with you, you can report it back. The kit has been used in case of even JN.1, when people started seeing that a lot of COVID primers were getting failed. They actually decided to either go for the shorter method or use the VSP. VSP kit actually helped them to come up with something that it is not the same variant that we are seeing previously. It is something new which is coming up. And then on the basis of that, the new primer was decided. The Arctic Primer 5, which we are seeing now for the COVID, is something that actually came out from as a result of research done using the virus surveillance panel, this particular panel, the first panel which was used. So there are multiple panels which are available with us. I'll share the details with you. Here, it is a use case when the researcher decided that let's look at the outbreaks and the antimicrobial resistance which is existing in a community. What they did is they took the sewage water, they used two different panels. One was the virus surveillance panel, which actually give you the snapshot of different viruses. Another was urinary pathogen panel that give you the snapshot of urinary pathogen and associated antimicrobial resistance. So the results were eye-opening. In case of virus surveillance panel, the routine techniques like PCR-based techniques were showing X number of pathogens circulating in the sewage, but using virus surveillance panel, because of sensitivity and its mismatch tolerance, it gave the 2x number of viruses. Looking at the antimicrobial resistance, so generally, in case of antimicrobial resistance, one has to do a lot of sequencing. People go for like 100 million reads in one go. 100 million reads is too much. In my first slide, I was talking about 3 million reads. Here I am talking about the 100 million reads in order to come up with certain specific antimicrobial resistance. In case of UPIP, they just need to do 1 million reads and they achieve what they were not able to achieve even with 100 million reads. So again, a cost saving and less cumbersome. How these are less cumbersome that I'll share. But first, let's go on to the virus. It is one of the very vast virus panel available in the market. It allows you to go for the sequencing of almost 200 viruses which are there in the WHO priority pathogen list. It is based on your these viruses can be captured using this panel. And most interesting thing is you can go for the whole genome sequencing of all these viruses. Now I'll share one very interesting case that I just done, and there will be a poster about it. So the team at BJMC found a case wherein it was ELISA positive using the it was a clinical sample ELISA positive for dengue, and they decided that okay, let's put it into this particular panel. Interestingly, ideally, this panel should give the whole genome sequence of dengue, right? But it gave the whole genome sequence of chikungunya. It was never dengue. It was chikungunya. So they would be presenting this use case over here. So the panel actually, of course, one cannot use this panel for the diagnosis purpose because you cannot sequence the whole genome for everything. But if your applications are that and you have an interest towards looking at certain mutations, what are the changing patterns that you are seeing? For example, in Pune, we witnessed that the chikungunya representation actually changed drastically. The patients complaining for the coming up with the chikungunya were having slightly different symptoms. So what were those things? They decided to sequence a lot of chikungunya using this particular panel. So National Institute of Virology again, when Nipah outbreak happened, they decided to go for this particular panel to look at what are the changes in the Nipah that has happened. If you're not having the sequence, whole genome sequence available for any pathogen in India, I think this is one of the things that probably you can use to develop the whole genome sequence. So you can look at the viruses, hepatitis, chikungunya, Nipah, dengue, rabies, almost entire breadth of viruses are covered. This is a pan-coronavirus panel. The panel actually covers coronaviruses from the different origin. It works on the same principle. There are probes designed across the genome, and you can actually capture almost these 200 different coronaviruses using this particular panel. RVEK, when I say respiratory virus enrichment kit, it is focusing only on the respiratory viruses, and you can achieve the whole genome sequence of these viruses using this panel. And these are some of the use cases coming from India and US, especially in India. It is Max Hospital along with the IGIB. They put the use of this particular panel in looking at the co-infections. And they found that beyond looking at the whole genome sequence of these viruses, they were able to even look at the co-infection profiles. And these are different viruses which are part of this panel. We will be sharing all these details with you so that you can have a look over the breadth of these panels. RPIP, and there is another one, UPIP. I would be sharing these panels in detail. Respiratory pathogen is having almost 282 different pathogens in it. There are probes for different pathogens which are engraved in this panel. This panel is also having almost 2000 plus antimicrobial resistance marker in it. So very effective panel when you are looking into we have seen several multiplex tests available in the market. This is again a culture-free method in which you don't have to culture anything. You can start from your bowel fluids or maybe sputum or anything, and you can look at these pathogens. Interestingly, now if I really look at anyone from FAS, Manjunath, can you please tell me that library preparation in case of enrichment, how much time is it going to take?
For
RPIP.
Okay. So for enriching, it would take maybe maximum 2 hours. That's all. You'll be using the probes and just capturing. You have to extract the DNA from the samples, and then you have to just hybridize the probes which are there in this kit, and then you have to capture it and elute it in the same way you have then we'll be adding data to by the end and sequencing it. So maximum 2 to 3 hours time.
Okay, so I'm going to stretch it a bit more, like let's make it like 5-6 hours. Okay, so we are working in a very leisurefull environment. So let's stretch it to 5-7 hours. Then sequencing. Now the sequencing can be done even at 4 to 5 hours. So in 9 to 10 hours, you'll get to know about all these pathogens which are like fungi, bacteria, viruses, and mainly causing the respiratory issues. And in addition to that, you will also get to know about the antimicrobial resistance associated with those pathogens. Coming on to the most crucial part, which is report. As soon as the sequencing is done, you can put it into the app, which is known as XT5. It comes along with the kit, and it gives you results in like 2 to 3 minutes time. So that's the entire time it is going to take, and we are really very committed to bring it next to the patient testing if it can be brought. As of now, the panels are research use only. Again, a use case coming from the Max Hospital Delhi, wherein they put the RPIP in use, and they cited that the panel works fantastically well and more sensitive and efficient in comparison to BioFire, which most of the hospitals really use. Coming on to the crucial part, if we really look at the costing, it is at par or even lesser than the BioFire. This is what you can achieve with NGS. Your platform, which is going to be there in your facility, can be used for the multiple purposes. You don't have to run one sample for one thing. You can run multiple samples for the different applications. Urinary pathogen panel. Again, this panel is 180 plus pathogen, but most importantly, this is the widest panel available in the market, covering almost 75% of the CARD database. CARD is having almost all the antimicrobial resistance marker linked with it. So it is 75% of that. We know that at any point of time there are many markers, rather pathogens would not be circulating in any environment, but it is having 75%. And again, it can be used on the wide range of biological samples. Importantly, it is culture free. What my suggestion would be, you can use it for the different applications. One of the clinician, very renowned clinician in India, and looking at the pathogens, we found that this can be repurposed even for sepsis. So that is a validation you can probably plan in your setup on your own. If such kind of planning can be brought next to the patient testing, it would be really helpful for you. But again, important thing is, you can do the analysis within the span of 2 to 3 minutes. And here is the summary of what I have mentioned so far. Will not be going into that detail. This is almost, we are approaching to the last phase. Discovery. When I say discovery, it is a hypothesis-free method. You don't know anything about what's there. So even your series sample is a discovery mode. But there could be a focus. I want to focus on certain specific pathogens. Then you can use the panel. But if you are really after something, because you are seeing in community there are certain infections you don't know anything about, you haven't seen previously, the discovery is the one way. And SCOPE2 actually happened that way. They started sequencing the samples. They did a shorter method in which they sequenced everything. They come up with something like whole genome. Then they designed the BRICA, which they started using and regularly for the genomic testing as well as the PCR-based testing. And post that, after identifying the spatial epitopes and all, they designed the vaccines. So this is how it works. This is a story. Maybe you can try to replicate it. So in Australia, this particular case of Hendra virus actually came into the picture. Hendra virus generally exists in bat, goes to horses, and the chances of going from the horse to human is very rare. But if it happens, there is 57% mortality which is associated with that. So what happened is, in one such case, the horse actually came up with certain symptoms linked to hantavirus, and they decided to euthanize this particular horse. With the carcass, when they put the PCR, they found that it was not the hantavirus. So what was that? At that point of time, they decided to go for the metagenomic sequencing, a discovery-free approach, and they found again that the virus has mutated. It was the time to come up with a new set of primers, and this is how they changed something related to the body factor. So this is one example, and last but not the least, again, because we have been receiving a lot of requests for the fecal matter transplant nowadays, and there is growing interest. Rather, because in our Ayurveda people say that your gut is your second genome. This is, we know from long while, and what we are seeing is that large number of people are now correlating the symptoms with a disease, and then they want to look at the microbiome. They are looking at the oral microbiome, they are looking at the gut microbiome. So that is one part of the story. They are trying to come up with certain set of biomarkers, and they are calling it biomarkers. These specific microbes which are existing in gut or maybe in oral cavities linked to certain specific, even cancer, CRCs. So this is an emerging area where the cancers are being linked with certain microbes. Now another thing, we have seen a lot of probiotics coming into marketing in the market and talking about that they are going to change your life effectively. But just like we eat a lot of supplements and they don't give a positive impact to us, the microbes do the same thing. They don't get colonized in our gut because they are not meant for us. It is very important to look at the gut. So that's why the techniques like metatranscriptome actually come into play, wherein you need to look at the functional diversity which is existing in the gut. So don't focus only on DNA side, but also focus on the RNA side. And this is going to help a lot when the probiotics are being designed. So there would be a next era coming up, which is precision probiotics, specifically meant for. So with this, I'll conclude my presentation, and I have shared about, the first part of this slide was the problems we were struggling with, and on the right side, these are the solutions that I have proposed just now. And thank you so much. Increase your beds further to the different hospitals. You can easily fill the flow cell. That's not a problem. You know that our country is a hotspot of outbreaks everywhere. It is everywhere. Looking at the dengue cases and chikungunya, and I have talked about the respiratory pathogen, urinary pathogen. These are a large number of pathogens. We are having such cases available with us. The filling flow cell is not a problem.
The last question, as in the case which you showed is it was positive for dengue as well, but actually it came to chikungunya. So there is too much technicality also involved in the case.
Yes.
So the only point which I wanted to ask is if suppose we are getting some pathogen seen in this, how much specifically, how much clinically you might have been correlated with the reporting also. Is it could be because of some technicality issue we may land up in mixing of the nucleotides and we getting detected some other pathogen which actually is not there. Because it's really an eye-opener, the LSS positive for dengue, it's coming positive for chikungunya.
So it was cross-checked multiple times. But what you are saying is right. The chances of cross-contamination always exist. It can happen even with a PCR. It can happen even when you are conducting a serological test. It can happen anywhere. But when someone is reporting, we are taking that it is error.
Many of your assays are culture-based, like respiratory and urinary. Is there any assay, culture-based method for blood, a culture positive broth, and then we do assay? How about testing directly from a blood sample from a sepsis patient, if you have a panel like this, or is it in the microphone?
So I would say such kind of panels are being validated for the different applications. For example, we talked about the urinary pathogen. we talked, then I showed it, how it can be used in the sewage sample. Nowhere we have validated that panel for the sewage, but people have validated it for the sewage. So similarly, there could be one approach wherein you can start working with a blood positive, culture positive isolate, and then you can actually go on to the directly, DNA isolated from the blood. This is something that can be done. Hypothetically, yes, it is possible, but practically, people are working on it. They are, repurposing this test for the different applications.
So then we might have to say DNA RNA extraction generally and take the whatever antigens at the end of it and then use it for sequencing line.
Yes, so the panel that I showed you is not the antigen panel, this is hybrid capture panel. But once you have the total DNA or nucleic acid available with you, you can do that.
In continuation of what you have spoken, sir, I would like to, for the sake of information of all these participants, because I stay in Pune, last few months has been very interesting observation, and you must have read in the newspapers that there were cases of dengue, there were cases of chikungunya, and the clinical picture was so varied that there was no adherence to the conventional syndromic presentation by these viruses. And only because of the RT-PCR and the NGS, there could be proper identification that there was a transgression on the part of dengue, like you mentioned a case that RT-PCR showed dengue, but subsequently it was detected as chikungunya. So these are variations which were observed. And what they were trying to correlate, like it happened in case of COVID. COVID had mutants,
yes,
and different waves, it has provided different type of mutants. They were speculating, like in case of dengue. Dengue is associated with platelet influencing factors and hemorrhagic manifestations. Now dengue started behaving with CNS manifestations. Chikungunya, known to cause skeletal-muscular disorders and this thing, started behaving like dengue, causing hemorrhagic manifestations in CNS, cases of cerebral hemorrhage and other activities. So these type of variations detected only because of the tool like NGS. So I will definitely compliment you for this presentation today. It's a wake-up call for all of us. And we must try to generate as much interest as possible to be after you to give us a solution in form of NGS, proper tool of NGS for infectious diseases, and furthermore the genomic sequences and further analysis. But it must be correlated with, I would rather add to it, if you use the artificial intelligence as a tool for surveillance, screening of the populations across the country, and then use the tool like NGS for further more analysis, and that will become a more apt contribution from this particular technology. Thank you.
Thank you. Thank you so much, sir. So I'll be around.
Finish with the data analysis part first. Carried our laptops for all the way for this thing and it's not happening.
Okay. Just 1 minute.
You can see here, share accepted. If you open this, it will take you to this page. So this is how the sample analysis, after completing the sample analysis, you are going to get a page something like this. Okay? The first two files we have already uploaded, right? Analysis is still undergoing. Once the analysis is complete, you will get a page, something like this, similar to like this, and in that you will have the report, analyze samples. Okay? You will also get a file named something like analyze samples. You click that. Once you click that, the reports will be with you. Okay. So are you able to come to this final page? This is where the report has been generated. You are able to see, right? Okay, summary report. Now, if you go down in the summary report, the most important thing here we look in COVID genome sequencing is the lineage. Okay, what lineage the particular virus is belonging to. Okay, whether it was Omicron, whether it was your Delta virus, those lineage information we want need to gather. Okay, so you can see here. For every sample, the lineage information is already given. Now, if you see the report initially, where we have uploaded 26 samples. Okay, so it defines that out of these 26 samples, 25 are actually COVID positive, right? Because one sample probably it failed to pass the QC. Okay, now all the reports of these 26 samples are now displayed in the table. If you see the table, you can see here the lineage information is also given, okay, which is much important from a biological point of view. Okay. Now, beside this, there are other columns where you can see the QC, whether it is passed or not, is also given. Right? And you can also see a column where the percentage of non and bases. It means around 99.72% of your bases has been like sequenced properly. Okay. Now, if it goes below 90%, sometimes what happens, it may not be able to identify the lineage. Okay. So this is a very good quality data where you are able to identify up to 99.75% of your COVID virus genome. Okay. You can even export this lineage information if you click the Excel sheet that is given here.
So if you accept the report as a good report, this non-N should be more than 90%. The cutoff is 90%.
Now, you will be able to detect the lineage there even. Means if you want to detect the lineage properly, means normally we need to have a percentage of about 90%. So if you see the Excel sheet that is given here, if you click it, you will be able to generate the report as an Excel file. If you go down, then there is another clade report related information. So this is going to tell to which clade the virus is belonging to. Okay, so we have 20B. If you look into the sample types, 20B, 20D. Different clades are there, 20B and D. And in the third column, the major substitutions or the genetic variation present in the particular genome and how it is helping to identify the clade. The information is given here. So the main important thing here is the number one lineage report. So it is through this lineage report during COVID time we have identified the Omicron strain, we have identified the Delta strains and like that. So the lineage report will show you whether it is COVID, Delta, whether it is COVID, Omicron, all those information will come up. Now if you want to collect, let us say, a FASTA file, okay, so individual virus genome FASTA files. Now if you see in the most of the databases, you can download the FASTA files of a particular genome. Now if you want to download that, you need to just download the FASTA files from here. Okay, and that particular FASTA files can also be uploaded to these two websites. The clade that is given here, if you can see here, right? The clade and the pangolin that is given here. The clade, next clade, and your pangolin COVID-19 lineage assignment. Now when you upload the FASTA file there, it will help you identify the clade and it will help you identify the lineage. Okay, this is just to cross check it, but the COVID pipeline has already identified that for you. Okay, so the lineage and the clade information is already being identified. But if you want to cross check it, what you can do is you can download the FASTA file of individual samples, upload in the next clade. You can directly upload here, and you can even upload here in the pangolin. So pangolin is going to give you the lineage information, and the next clade is going to give you the clade information of the virus. Okay, so sometimes during the COVID, when ICMR was performing this genome project, the individual COVID genomic sequencing centers, they need to upload the clade information as well as lineage information. Okay? So even the FASTA file information also. So what they have done is, after performing the genome analysis, genome sequencing, they have downloaded all the files from here and they have submitted it to the ICMR portal. Okay? So this is how it is done for simple COVID genome sequencing.
That is actually the FASTQ will contain the quality information of the sequence. But FASTA will not have. FASTA will only have the sequence and the head.
Yes, we are taking over there. Actually, due to medical emergency, she will not come. She could not come. So you have already taken it? Yeah, we have. Yeah, this is the same thing that we have done. Okay, fine. So after this, we are the. Yeah, we are the. Fine. More time more? It's almost over. Then we go the. Yeah. What is time? 5:50. Okay. Take another like 15-20 minutes. Right.
So we need to get that language, we have to understand that. So we had planned it that way, that we have a second day on data analysis, but we could not do anything. The election came in, we had to squeeze our program to a one-day program, and we tried our best to give you that overview or
I think these people are there for tomorrow also. If there are some leftover matters, they can discuss it individually with them.
Anytime.
Tomorrow, there are two faculty there. Tomorrow.
Till Saturday.
Yeah. So anybody can get the inputs and interact with you individual basis at some point of time.
So we have a stall, so we will be there.
I think that should be easy matter. I mean, you can't grab everything into one session.
And also we have that WhatsApp group. We'll keep that group active. Anytime you feel like you have any difficulty, please feel free to put your message. Anil is there in the group. The other people from Premas as well. The entire team is there. We are there. If possible, from our side, we'll be contributing to things.
Keep the group viable for another 10-15 days.
Sure.
In the meanwhile, people will soak in the information whatever they have got. They will raise some queries. They put it in the WhatsApp group and then just let these people answer and interact for a period of time. I think that should be fair enough.
Yes, sir. We'll keep that group open. We'll not close
Pretty
that.
viable for 15 days. You can soak in all the information, read through what they have given, plus read some extra also. Go back to your individual laboratories and find out if there are any more queries and come back on the WhatsApp.
Yeah, we understand this is not a one-day program and just doing it for a couple of things, working on demo data does not give you a complete experience of NGS. You need